Hi everyone, welcome to Medical Microbiology. Today we will discuss chapter 11, which is about the physical and chemical control for microbial growth. So for the first lecture of this chapter 11, we mostly will talk about the physical methods. Okay, so physical methods in lecture 1. So before we go to the discussions about the physical method to control microbes, we have to know some terminology on this chapter. So the physical and chemical controls, usually we use this methodology to control bacterial or microbial growth outside our body. Outside our body. So mostly this is for prevention. Okay, for example, when we clean our bathroom, we clean our floor, this is a type of this control, physical or chemical control, and which is the preventions against some microbes that might cause disease to us. And also when we wash our hand with the antiseptic soap, this is considered also as a prevention, and this is going to be chemical control of microbial growth. Okay, so this is chapter 11, physical and chemical control of microbial growth. And again, mostly this is for prevention. Now, some terminology. First one is disinfection. Okay, so what is disinfection? This is the destruction or removal of vegetative pathogens, but not the endospore. So when we talk about disinfection, it is actually remove some bacteria or viruses, but not the endospore, because the endospore is really difficult to remove or to destroy. It's not enough using this disinfection. And disinfection, they are applied on the inanimate object or non-living object. Okay, for example, we use Lysol, Clorox to clean the bathroom. This is considered as disinfection. Sterilization, this is the method that is used for complete removal of any microbial type. So including including the endospore over here. Right? So it will remove all type of microbe, including the most difficult one to remove, which is the endospore. And of course, this is going to be used on inanimate or non-living object. Antisepsis or antiseptic, this is the chemical that used on living object, okay, animate object. Like for example, when we use hand soap, okay, antiseptic soap, this is considered as an anti-sepsis method or antiseptic method, to destroy, inhibit mostly vegetative form. It is not killing or destroying the endospore, because endospore is the most difficult type of microbial form to be destroyed. And again, this one is going to be used on animate or living object for the antisepsis. Sanitation, this is any cleansing method to remove microorganisms from inanimate object or surface for non-living surface to reduce the potential for infection and spoilage. Okay, and then degermination, this is the one that is used in living tissue to remove bacteria on living tissue, and usually this is done by doctor when they try, or they're ready to perform surgery, for example. They will do this process, degermination, which is removing bacteria or any microbial type from their hand or from their part of their body. So it will not transfer the microorganism to the patient body, okay, when they perform the surgery. Microbicidal agents. So first, this side over here is mean kill. Okay, so therefore microbicidal agents mean the agents that kill the microbes. And the names will be depend on what we use for. Like for example, if we use to kill bacteria, then it is called as bactericide. If we use for kill the fungi, it's fungicide. If this is for virus, virucide. If it is used for endosperm, okay, endospores, that's called the sporicidal or sporicide. So this is the chemical that kill bacteria. Okay, so bactericide over here is only kill bacteria, but not really kill the endospores. In order to destroy the endospore, then we have to use the highest type of agent, is called the sporicidal. Okay. Microbiostasis. So stasis mean stay the same, which is the agent that used to prevents the microbial reproduction. It's not really kill. So this is not really kill. This is just prevent reproduction, not killing the bacteria. Example is when we put our food inside the refrigerator, the lower temperature in the refrigerator is not really kill bacteria on our food, but it will prevent or reduce the reproduction of bacteria on that food. So therefore, food can be fresh for several days. Compare if we put outside the on the room temperatures. Okay, so again, microbiostasis is not really killing bacteria. It will reduce or prevent the reproduction. If you want to kill bacteria, for example, then we have to use bactericide or microbiocide. Okay, these tables again, some terminology. Decontamination. This is the destruction or removal, reduction of number of undesirable microbes. So the methodology may be asepsis. A mean no. So sepsis mean bacteria. So it means that we remove bacteria, no bacteria. Okay. Disinfection. This is also decontamination method. Sanitation, degermination, sterilization. This is considered as the decontamination type. Sepsis means growth or bacteria or microbes. When we talk about sepsis, mean there is a growth. There is growth of microorganism in the tissue, the body tissue. Okay, so the presence of infected bacteria in the infected wound, for example, maybe in the blood. This is what we call the sepsis. Okay, the growth of microbes in the tissue. The opposite of sepsis is going to be asepsis. So A actually means no or not. So no or not. So asepsis means no growth, no bacteria. So no bacterial growth in the tissue. Like for example, when we clean the skin with iodine, it might make the tissue become asepsis. And remember, the Joseph Lister promoted this aseptic technique in medical place. So this is called the aseptic asepsis. No growth, growth, no bacterial growth. Antiseptic, so this is gonna be the chemical used for removing some bacteria, and it is applied on living object, like on our body and our skin, for example. So this is antiseptic. Usually, you know, for example, using iodophor, antibacterial soap, chlorhexidine, this is considered as an antiseptic. Disinfection, this is very similar with the antiseptic, but it is used on non-living object. Okay, like for example, using bleach, boiling water, Lysol, Clorox, that's considered as disinfection. We apply these chemicals on non-living object. Okay. But again, remember, this infection will not remove the endospores. Sanitation, this is any cleansing method to remove bacteria from non-living object, okay, inanimate object or surface. Dishwashing, laundering, for example, this is considered as sanitization. Degermination, this is the one that is used on living object, on living tissue. Okay, like for example, I gave you example already, you know, when the doctor want to perform the surgery, then they will perform the degermination. Okay, so clean their hand with several type of chemical that will able to remove bacteria or microbes from their hand. And sterilization, this is the destruction of all viable microbes, including endospore, which is the toughest type of microbial form to be controlled. So endospore, you know, we have to remove, we have to destroy them with the process called the sterilization. Example of sterilization is autoclave, ionizing radiation, okay, incineration. This is also sterilization using the gas, gas sterilization, like ethylene oxide. This is also sterilization. Okay, now we're gonna focus in this, what we call the target. And of course, we know what is our target. What are our target to be removed? Okay, of course, gonna be pathogenic microbes, like for example, bacteria, okay, fungi, some protozoa that can cause disease, worms, viruses, and prion. So these are our targets. When we use chemical or physical methods to remove, what we remove is, we try to remove is actually this type of microbes. This is what we call the target for our physical and chemical control. Now, from that target, then we can classify them into 3 different groups. Our targets can be classified into 3 different groups. The first one is the highest resistant type of microbes. So this is the highest resistant target, which is prions, bacterial endospores. They are really difficult to remove. Bacteria that produce endospore, example is Bacillus and Clostridium. So it's not many type of bacteria that produce endospores, only several types, including Bacillus and Clostridium. And again, endospores are considered as a form of bacteria that is really difficult to control. So it's going to be highest resistant type. The second ones will be the moderate resistant target. Including in here is the cyst of protozoa. So some protozoa are able to produce cysts. Usually if their condition is not optimal, then they will not reproduce. They kind of cover themselves into additional membrane, making what we call the cyst. So this cyst is kind of difficult to destroy. So they are in the moderate resistant. Most of non-enveloped virus or naked viruses, they are also moderate resistant. Several bacteria, even though they do not have endospore, that they have special wall, okay, resistant wall that make them difficult to control. So include in this type of bacteria are Mycobacterium. So this is the one that can cause the TB disease, and also leprosy. So Mycobacterium is difficult to control. Pseudomonas, Acinetobacter, Staphylococcus. You see this one. So this also moderate resistant type of microbes. And the last one, of course, the one that easy to be controlled or easy to be destroyed. So most of vegetative type of bacteria, they are easy to remove, to destroy. So using, for example, disinfection, antiseptic, maybe kill this type of bacteria. But maybe, you know, some antiseptic will not kill Staphylococcus because they are more difficult. But of course, they will not kill the endospore because it's really, really difficult to control. But some antiseptic, disinfectant, they mostly will remove this least resistant type of microbe, including the molds, the fungi, yeast. Most of the envelope viruses, they are really easy to control. Like, for example, flu viruses actually is really easy to destroy when they are outside our body. Washing our hand with the antiseptic soap, for example, will kill these flu viruses, okay? Because they are envelope viruses that easy to destroy. And of course, the protozoa tropozoid. Tropozoid means a form of protozoa that they reproduce. So this is the reproductive type of protozoa. So usually, you know, if the condition is good, then protozoa will be in this type of condition. It's called the tropozoid. If the condition is not good, then some of them able to make cysts. So when it is cyst, it's gonna be more difficult to control. But if it is tropozoid, then it's gonna be easier to control. Now, this is experiment actually comparing between endospore and vegetative form of bacteria. So it will compare which one is more difficult to destroy. So if we use, for example, autoclave, moist heat over here, it requires at least 120 centigrade to destroy endospore. Only 80 centigrade, okay, which is lower than the boiling temperature, to kill the vegetative form. So boiling your food, for example, mostly will kill the vegetative form of bacteria, but it will not kill the endospore. So if we compare between this vegetative form and endospore, we see using the heat, moist heat. So the endospore is actually 1.5 more difficult to control. If we use radiation, for example, X-ray, if it is endospore, it requires 4,000 dose. If it is vegetative form, only 1,000 dose. So it's 4 times. It's 4 times more difficult for endospore to kill, or 4-time dose are needed for killing endospore. If you're using sterilizing gas like ethylene oxide, then endospore require 1,200 milligram per liter, while the vegetative form only 700 milligram per liter. So about 1.7 time, okay, dose, or more difficult. Look, when we use chemicals for removing the endospore, it's called the sporicidal, like glutaraldehyde. In order to remove endospore or kill endospore, it requires 3 hours application. For the vegetative form, it's only 10 minutes enough to remove them or to kill them. So this is about 18 times for endospore compared to the vegetative form. So again, this is showing us the endospores are more difficult to destroy compared to the vegetative form of bacteria. Now, when we talk about removing or killing bacteria, then we talk about the microbial death. So this is a permanent loss of reproductive capability, even if it is under optimum condition. So it means that even though they are in the optimum condition, if we put chemical, for samples on them, then they will not be able to reproduce. They will die. Okay? So that's considered as a microbial death. There are some factors that affecting the microbial death. Of course, the first one will be the concentration of the microbe itself. The higher the concentration of microbes, then the more doses needed. Right? The more doses of chemical needed to kill them. Right? The nature of microbes. Remember, there are microbes that is higher resistant, moderate resistant, or least resistant. Right? The temperature and the pH of the environment, the concentration and the dose of the agents, okay, and the mode of action, and the presence of solvent, organic matter, or inhibitor. This is, for example, water treatment company. They usually will put more dose if the water have a lot of organics matters in that water. Because what? Because the organic matter, sometimes they will combine with the chemical agents that is used for destroying microorganism. Therefore, it's going to be less effective because this organic matter actually bind to the chemical used for bacterial control. Okay, so if we see this, so this is the general rule. Okay, so if we have a lot of bacteria in here, then it's required time to kill them. Okay, until this. So this one is no bacterial growth. So let's say it's about 13, 14 minutes to sterilize this dose. Okay, so if we compare the higher dose, the higher load with the low load, then the low load will require less time. So maybe about 8 minutes. If it is higher load, maybe about 15 minutes, for example, to remove them. So the more concentration of bacteria, the more time needed to control them. If it is vegetative cell, compare with the spore, of course, vegetative cell will require less time. Let's say maybe only 10 minutes over here. The spore maybe take hours. Okay, takes hours in order to remove them. So spores are more difficult to remove or destroy compared to the vegetative cell. This is the general rule. Okay, if we do not give any chemical agents or physical agent, for example, the bacteria will keep growing over here. So growing. But when we put chemical here, for example, then the bacterial growth will be going down. So this is the time when they start to going down, when we apply something over here, whether chemical or physical type of agent. Now, practical concern, so especially if you have a business or a dentist, for example, they have to think about it, whether they need to remove, bacteria, they need to perform sterilizations or not. Okay. First, we have to know whether the application or the equipment require sterilization or not. Like for example, dentist equipment, mostly they are very expensive equipment. Therefore, mostly they will be, they required sterilization. Because what? Because they will be reused. So the dentist will not throw away their equipment because that's expensive. If it is disposable equipment, for example, or disposable thing, then they do not require the sterilization. And also whether the item can, withstand the heat, pressure, radiation, or some chemicals. So for example, plastic maybe will not withstand the heat, so we cannot, for example, use incineration for plastic type of equipment. Is the method suitable? Is the agent penetrate into the into the equipment? Whether cost and labor efficient or not? Okay. Now, when we talk about the target before, so our target, remember, bacteria, viruses, fungi, prions, protozoa, worms, for example, that's the target. But within the target, there are specific structure. So this one, when we talk about target over here, is actually the structure. Okay, the structure of the microbe that can be destroyed by the physical or chemical methods. Okay, for example, so when we talk about bacteria, remember bacteria, it is a cell that has cell wall made of peptidoglycan. And then under the cell wall, bacteria has cell membrane. Okay, so this is cell wall, this is cell membrane, and then inside them there is a cytoplasm, and there will be chromosome. Remember, bacteria have chromosome, but they do not have the nucleus. So this is the DNA, okay, chromosome or DNA. And bacteria also have the structure that produce protein. It's called the ribosome. Okay, so these are the structure of bacteria. So if we see the structure, then each of the structure can be a target for the chemical or physical control. So the target will be what? The first one, the cell wall. Okay, so there are some agents that actually able to destroy the cell wall, the peptidoglycan. For example, detergents, alcohol, they can destroy the cell wall. So this is the first target. It's gonna be. The second target will be the cell membrane. There are some chemical agents that actually destroy the cell membrane. They are called as surfactants. Many antiseptic soap, they are surfactants. The one chemical that destroy the cell membrane. Now, nucleic acid and the ribosomes will be the next target. So the protein is made in the ribosome. The nucleic acid will be the, in the chromosome. So this is going to be the next target, which is the target on the nucleic acid and the ribosome. And also, after the ribosome produce the protein will be available here. Some protein like enzyme, for example, used for chemical reaction. Therefore, there are some chemical that use that protein function like enzyme function as the target. So it's going to be the next target over here, the protein. Enzymes is also type of protein. So these are the structure, the target that will be used by physical or chemical control. Okay, now we're going to see what kind of methods used for controlling bacteria. So we're going to classify them based on the method or the agent that we use. If it is physical, then we call it a physical method or physical agent. If you use chemical, then it's going to be chemical method or chemical agent. If it is mechanical, then we call that as a mechanical methods. And usually physical and mechanical, they are very similar. In many books, they actually consider as one method, physical and mechanical over here. Okay, so therefore it's actually only two classes: physical mechanical agent and then chemical agent. So we will discuss these physical mechanical agents in the first lecture. Chemical agent will be on the second lecture. Okay. Example of physical agent is using heat, okay, or temperatures. Okay, maybe hot temperature or maybe cold temperature. That's considered as a heat or temperature. So heat can be dry heat or maybe moist heat. Okay, moist heat, okay, including steam underwater like under pressure, steam under pressure like sterilization, or maybe boiling water, hot water, pasteurization. If we use the steam under pressure, mostly it's going to be sterilization because it will remove the endospore also. If we just use pasteurization, boiling water, mostly just disinfection. Do not really remove the endospore. For dry heat, incineration and dry oven, like oven we use in the kitchen, this is considered as a dry heat. Most of them is sterilization. It actually kill or destroy the endospores also. The next physical one is radiation. And for radiation, there are two types, ionizing and non-ionizing. For the ionizing, usually using the methods that end with ray, like X-ray, gamma ray, cathode rays. That's ionizing. And they are sterilization. They can kill and destroy the endospore. If it is non-ionizing, usually end with light, like UV light. And they are going to be disinfection only. Do not remove or destroy the endospore. For the mechanical, mostly filtration, air filtration. Like for example, we have the air filtration in our house. We have filter for our air. Then it is disinfection. It's not really sterilization. Oh, don't forget, if you have filter in your house, you have to change it. Some filter maybe it has to be changed every 1 month or 3 months. It depends on type of filter that you use, the air filter that you use. Don't just use the same filter for years. It's actually causing problem to your air. air in the house. It may be more bacteria actually if you do not replace the air filter. Liquid usually also can be filtered. So maybe some people use filtration in their house. You can buy the water filtrations in Walmart, for example. That's actually filtration. Many of them is actually sterilization. Okay, many liquid filtrations are sterilization. This one is going to be in this lecture, chapter 1. The second lecture will be chemical agents, maybe liquid. This is the one that mostly used in many places, including in the household. So we use liquid, a lot of liquid, Lysol, Clorox, antiseptic soap, and etc. So if it is liquid, if it is used in animate object or living object, it's called the antiseptic or antisepsis, like hand soap, for example, or antiseptic soap. If it is used on the non-living object or inanimate object, then they can be disinfection, like Clorox, Lysol, or maybe sterilization, like glutaraldehyde. That's a sterilization. The second type will be gases. Some gases used for sterilization, like ethylene oxide, and some gases may be just disinfection. So this the methodology that used for bacterial control. Now, if we compare the moist heat, so moist heat and dry heat, then actually the moist heat is better, is more effective. So if we compare here, however, for moist heat, in order to kill or remove endospore, is require only 120 centigrade temperature, and then take about 15 minutes. Compared to the dry heat, if we use the same temperature, 121 centigrade, then it's require almost 600 minutes. Okay, so see that one is almost 10 hours. So that's compare if the moist heat with the dry heat. Moist heat is more effective. And then if we increase the temperature 125, then it's only require 10 minutes to sterilize the equipment. If it is 130 degree, so it's only required 3 minutes. So for moist heat. So moist heat, actually the lowest temperature that we use is 120 for 15 minutes. That's the lower temperature used. Now look at the dry heat. If 140 is 180 minutes over here, almost 3 hours. If it is 160 degree, then about 2 hours, 120 minutes. If it is 170 degree, it's require 1 hour or 60 minutes. So again, if we compare between moist heat and the dry heat, the moist heat is more effective. Now, when we're talking about comparing things regarding the temperature, so there are two types, TDT, which is thermal death time. It means that the shortest time required to kill the test microbe at specific temperature. Like, the temperature will be the same. Let's say it's using 121 centigrade before. So when we're comparing moist heat and the dry heat with 120 centigrade, for the moist heat only require 15 minutes, but for the dry heat is require almost 6 hours, or even maybe 6 hours more. And then if we talk about the TDP, lowest temperature at the same time. So for the 10 minutes, for example, for the moist heat is require less temperature compared to 10 minutes using the dry heat. It's require more or higher temperature. Okay, this is the example of method that's widely used for sterilization. Sterilization using the moist heat or steam under pressure. So the most common one is using autoclave. So autoclave is used in many places, clinics, hospital, including in my lab. I also have autoclave. So because why? Because it's very effective to kill or destroy bacteria, including the endospores. Okay, and then there is another method, non-pressure steam, like tindalization. This is going to be less effective compared to the autoclave. Boiling water is going to be just disinfection. It's not sterilization. The endospore will stay, will not be destroyed. There is another technique, it's called the pasteurization. So this is moist heat type of methods, okay, physical methods. And usually this is applied for food, okay, like for example, milk, okay, juice. Okay. This is the type of methodology that used to remove some microbe. And remember, this is not sterilization. Okay? So this is not sterilization. So if there is a endospores on that media, for example, that food, then it will not be killed by this pasteurization. This is the picture of autoclave. So you have the chamber over here. So we put the equipment or things that we're gonna clean, sterilize in this chamber. There will be water, which is moist, and also the pressure applied to this chamber. Okay, that's moist heat. Now we go to the dry heat. There are several methods. The most method used is incineration. So many labs will have this small incineration, like electric heating coil. Okay, sometimes using flame, also use gas flame, for example, in many lab, like a chemical lab or physical lab or biological lab, they have the gas flame, for example. Dry oven is also type of dry heat. Yeah, this is type of small incinerations mostly in biological lab, like microbiological lab, in order to sterilize this loop, okay, transfer loop. So here is actually hot or heat that will kill the bacteria, including the endospores. Okay, so again, this is just summary of the technique that we use for heat, whether moist heat and dry heat. So moist heat, usually using steam, boiling, hot water. If it is with pressure, it's called the steam under pressure. Like example, using the autoclave, 120 degree Celsius needed, minimum, with 15 minutes to 40 minutes. So the lowest temperature will be 121, and the lowest time will be 15 minutes. Tindalization is actually using lower temperature, but it will take more time for this under pressure steam. The next moist heat is pasteurization, and again this is used for disinfection of milk, dairy products, juice, for example. It can use lower temperature. Therefore, this is just disinfection. Okay, this is not sterilization. Boiling water, so usually 100 degree, and this is mostly also disinfection. It is not sterilization. So endospores will not be killed by boiling water. Now, dry heat, again, dry oven, it's gonna be sterilization. Okay, so this is type of sterilization. Also incineration, this is a type of sterilization. Again, if we compare between the dry heat and moist heat like autoclave, then autoclave is more efficient. Okay, now another heat, another temperature is called cold. Like for example, refrigerator or freezer. That's cold type of method. So this is as I mentioned before, this is microbiostatic. It is static. So what does static mean? Prevents reproduction. So it's actually not really kill the bacteria. It will prevent the bacteria for growing. But if we put them outside this cold temperature, then the bacteria will going back to the normal reproduction. Okay, so again, cold is not biocide. This is just microbiostatic. Desiccation, this is removing the air from the chamber or from the area. And usually cold and desiccation used together with the method is called the lyophilization, which is freezing, drying at the same time, and then also removing the suctions of air from the equipment. And this is preservation. And again, preservation means biostatic, microbiostatic. It will not kill the bacteria. Okay, so the next physical control will be radiation. Okay, after heat, then is radiation. There are two types of radiation, ionizing and non-ionizing. Again, ionizing usually using rays like gamma ray, X-ray, cathode rays. This is a type of ionizing radiation. And this is going to be sterilization because it will actually destroy any type of microbes, including the endospore. If it is non-ionizing, they usually using light like UV light, UV light radiation. So if it is using rays, it's going to be sterilization, which is ionizing. If it is light like UV light, this is non-ionizing. This is just disinfection. It will not kill the endospore. Okay, this is visible light, the light that we see, we know. So if it is like, the white light that we have, that actually they have several spectrum. You can see the spectrum during the rain, like you see the rainbow color. This is actually the spectrum of visible light. And this spectrum usually safe for us or for microorganisms. They will not kill the microorganism with this visible spectrum of light. But if we lower the light, then it becomes something that have energy. It can kill bacteria. This is actually higher power, and mostly this over here, this is going to be ionizing. It's a sterilization kill and destroy bacteria. This is ultraviolet, UV light is closer to our visible light. This is can be used as disinfection. Okay, X-ray, gamma ray is gonna be used for sterilization. So microwave also can be used to heat or to kill some microorganism. But again, this one is gonna be the highest energy over here, because it's shorter wavelength, gonna carry higher energy. The more energy, then gonna be the more power to kill bacteria. So therefore, they can be used for sterilization. The one that closer to our visible light, like UV light, they still can be used to destroy some bacteria with disinfection or non-ionizing radiation. Again, this is ionizing using X-ray, gamma ray. It's gonna be actually it's breaking the DNA. So the target is actually the DNA of the microbes. It destroys it. If using UV light, non-ionizing, it's not really destroy, it's just change a little bit. So the bacteria, some of them actually still be able to live. Mostly the endospores will not be destroyed by this UV light. And also UV light usually cannot penetrate some solid object. For example, in the UV light room, if we wearing the clothes, usually we are safe from this UV light. Or if we are under UV light, mostly it will not really change or destroy our cell. But if it is X-ray, for example, or gamma ray, it's gonna be not only killing the bacteria, but also causing some problem to our body, to our cell. So therefore, if you have an X-ray, they usually cover your body with some very solid materials, okay, in order to protect against this X-ray or maybe gamma ray radiation. Okay, this is just some application for radiation, for ionizing. And again, this is sporicidal. Sporicidal means it kill endospore, destroy the endospore. So this is sterilization. Okay, so this is used for some equipment maybe in the medical or in the hospital. UV light, this is just it's not really sterilization. This is just disinfection, like some germicidal lamps, maybe used in some room in order to protect against some bacteria. So it's kind of a little bit sterilize the room with the UV light and sterilizing from some type of bacteria. That's for the radiation. The next physical, this is actually, in our table before is mechanical, right? So this is mechanical type or methods. So filtration, water filtration, for example, liquid, using this filter paper. Sometimes you can buy the water filtration stuff in Walmart, for example, and put it under the faucet, or maybe you put it on the drink container. So that's actually filtration. So it will filter some bacteria, okay, maybe including some dust. Okay? And I think they should, in the book, they don't really put the other filtration, which is uh, this is the first one over here is, okay, water filtration, and second gonna be air filtration. Okay, so again, in your house usually you have this air filtration system that you have to replace the filter maybe every month, every 3 months, maybe. It depends on what type of filter that you use. That should be enough for lecture 1, chapter 11. We will continue with the next lecture, which is chapter 11, lecture 2, which is the chemical methods. I see you the next lecture. Goodbye now. Hi everyone, welcome to Medical Microbiology. Today we will continue chapter 11. It's going to be the second lecture, and it will talk about the chemical agents used for microbial control. Remember this classification for the agents or the methods, okay, physicals and mechanicals, they are combined in one group. We already talked about these physical and mechanical methods in lecture 1. So today is lecture 2. We will discuss about these chemical agents. So chemical agents for microbial control. So it may be disinfectants. This is used on non-living object. Antiseptic. This is chemical used in living object. Sterilant, there are some chemical that use as a sterilants. This is on non-living object. Degermers, this is on living object, like for example, doctor that want to perform surgery, they usually use degermers. And there are some chemical used as preservative, like in the food, preservative. Some people put the chemicals on it. So some desirable qualities of germicide, rapid action with low concentration, because higher concentration usually more expensive. The solubility in the water or alcohol. Broad spectrum is mean that that chemical able to kill different type of microbes with low toxicity to human, to us. Penetration to the surface, non-corrosive, especially if we use in our equipment, and non-staining, like for example, if we use in some place or area that require that place stay the same, the color will not change after chemical application. And of course, affordable, cheap, and ready to be used. Okay, just for information, the chemicals method or chemical agent is actually the most common type of agent that used for prevention of bacterial growth. So this is the most common type of method used in many places, in the house, hospital, schools, and many places, they usually use these chemicals. So this chemical agent can be classified into 3 types. Just remember when we classified the targets, the bacteria, viruses, is also classified into 3 types. Okay, the highest resistant, intermediate resistant, and the least resistant type of microbes. So the same thing with the chemical. There are also 3 classes, which is the highest the high level. It means that this is the one that can kill endospore. It can be used as a sterilization or sterilants. The second one is gonna be the intermediate level. This is the one that may be used for destroying the intermediate type of resistant bacteria or microbes. And the last one is the low level. This is the one that used for least resistant type of microbes. So mostly like antiseptic in this low level type. Because what? Because antiseptic used in also in our body, in our skin. So we will not use higher level of chemicals on our skin. Okay. So high level, intermediate level, and low level. The high level will kill the endospore. So it's used for sterilant or sporicide. The intermediate level usually used for some intermediate resistant bacteria, and low level is used for least resistant bacteria or microbes. This is some agent that used for cleaning or controlling the microbial growth. Like chlorine, for example, many of them is sporicidal, can be used to destroy many type of microbes. This is going to be intermediate level. It has toxicity, so sometimes can irritate our skin, or maybe if it is gas, producing gas that may be toxic into our body. Iodine, this is also intermediate level. It can be used as sporicidal. Phenolics, this is some of them low, some of them intermediate. This is really depend on the concentration or dose. Chlorhexidines, mostly used for bacteria, fungi, viruses. Some low, some intermediate level. And again, it depends on the dose, the concentration. They are considered as low toxicity to our body. Okay, alcohols, maybe this is the one that we use most of the time to clean or disinfect some area, or even clean part of our skin or body, for example. Mostly used for bacteria, viruses, and fungi. They are intermediate level. It is toxic if it is ingested. Okay. Hydrogen peroxide, this is also we use a lot. They are considered as sporicidal. Some of them high level, used as a sterilant, especially if it is high concentration, but they are also toxic to our body. Toxic is ingested or toxic to our eyes. Quaternary ammonium compounds, mostly used in soap, antiseptic soap, for example. This is used for bacteria, virus, fungi. Mostly they are low to intermediate level. Soap, antiseptic soap, they are very low level. Because what? Because we use it in our body, so it has to be low level type of germicidal. Some mercurial agents, this is also low type of germicidal. Silver nitrate, mostly used for bacteria. They are mostly low level. Glutaraldehyde, this is one of the sterilant, liquid that used for sterilization. So therefore, this is sporicidal. They can be used for removing or destroying endospores. So make this. This is usually used for sterilization. Orthotaldehyde, this is also sporicidal, but some of them is intermediate. into high level. Ethylene oxide, I'm gonna give you the sign also. This is gas that usually used as a sterilant. So it is sporicidal. They can kill or destroy the endospore. Dyes, some dyes is used for prevention also, but they are very low level of germicidal action. Okay, so again, this is the category of chemical that used as chemical control or germicidal. Right? A group of halogen, phenolic, chlorhexidine, alcohol, hydrogen peroxide, aldehyde, gases, detergent and soap, heavy metals, dyes, and some acid and alkalis. Now we start with the halogen. So halogens, including the halogen is chlorines, iodines, and fluorine also over here, but usually fluorine has a very high toxicity to our body. So less use compared to chlorine and iodine. For example, chlorine, chlorine bleach like hypochlorite is used for water, water disinfection. So many water treatment uh, place using this hypochlorite, or maybe this hypochlorite will be used to uh, in swimming pool, for example. Okay, that's a hypochlorite. Iodines, like iodophore, this is also halogen that used for sometimes disinfection and sometimes also for antiseptic, like betadine, for example. This is antiseptic use on our skin. This betadine and iodophore is considered as intermediate levels of chemical agents. Okay, so chlorine, chlorine gas. This is disinfection in drinking water, for example, use chlorination. Hypochlorite, this is also for chlorination. Okay, common household bleach, for example. Chloramines, this is also chlorine type of chemical used as alternative for chlorine in treating water supplies. So in many bigger water treatment plant, okay, they usually use chloramine because chloramine is considered as safer compared to the chlorine. So, but this chloramine is kind of a little bit expensive, to use compared to the chlorine. Just for example, in Lexington, they use chloramine in their water treatment plant. But for some water treatment plant in Eastern Kentucky, like Pennsville, for example, they still use the chlorine. So chloramine is considered safer compared to the chlorine. I'm going back to the previous PowerPoint over here. I just want to show you why the chloramine is considered safer compared to the chlorine. So if you use chlorine, usually there is a byproducts, like this one over here, 3 halomethanes, and also haloacetic acid or HAA. It is the byproduct of using chlorines in water treatment plant. And these two chemical, THM, 3 halomethanes, so 3HM, and haloacetic acid HAA is considered as carcinogenic. Okay, it can cause cancer after long term of use or long term of contamination in our drinking water. So when water treatment use chloramine, they usually will produce this very little concentration. Eh? But if the water treatment use just chlorine, they produce more of this THM and HAA compare the water treatment that use chloramine. Iodophores like betadine, for example, this is one of the antiseptic. This is most common use for skin and mucosal membranes. Okay, it's used also for surgical hand scrub, which is cleaning the hand of the doctor before performing the surgery. There are some elemental iodine that use also for disinfection of plastic instruments. Remember, plastic instrument you cannot use or put them in the heat equipment and the heat methods. Yeah. Okay, now the second type is phenols. So maybe phenol like carbolic acid or chlorhexidine. So the chlorhexidine is one of the most type of chemical use in some hospital, for example, used for degermining agents, so for degermination. So usually chlorhexidine used a lot by the doctor that want to perform the surgery. Phenol also used in many cleaning product like Lysol, for example. So they also use phenol as active ingredients. Okay, so this is again still in the phenol type, phenolic type of chemical agent. So maybe creolin, bisphenol, hexachlorophene, and triclosan. So this is a type of phenolic agent, including the chlorhexidine, also in the phenolic type of chemical agent. Okay, so this is chlorhexidine. Again, this is, I just give you the sign, this is used in hospital for degermination. Okay, the third type is alcohol type of chemical agent. Okay, they can be disinfection or also antiseptic. Okay, so concentration of 70 usually is actually more effective compared to the 100% of alcohol, because 100% of alcohol is very dry. Sometimes it's not penetrate the place that to be clean, so therefore it's not that effective compared to the maybe 70% of alcohol. Yeah. The most common one used is in medical field is the ethyl alcohol or ethanol. And for household, usually using the isopropyl alcohol. So mostly you will not find ethanol now in Walmart, regular place. It's required mostly, maybe prescription in order to use ethanol, because ethanol can be used also for drinking, for drunk. But the isopropyl alcohol, it has a very bad smell, so it's not really used for some people to drink it, and it is also considered as more toxic to human than the ethanol because it has, you know, bad flavor compared to the ethanol. Okay, so this is just example of nurse using alcohol to clean equipment or maybe clean their hand. Hydrogen peroxide, this is the next type, the fourth type of chemical used for controlling agent. Many of them is used for example, cleaning the contact lens or also for cleaning the equipment. And if it is low concentration, sometimes it can be used to clean our mouth. The next group is aldehydes. Glutaraldehyde, for example, this is sterilant used for some instrument that cannot be put in the autoclave or in the incinerations. Okay. Formaldehyde. Formalins is the liquid type of formaldehyde. So this is a liquid or aqueous solution. This is intermediate to high level of disinfectant. Gas, and this is the next type of chemical use. Some of them use as sterilization, like this one, ethylene oxide. Okay, this is the most common gas sterilant use. And usually if using the ethylene oxide, we have to put the equipment inside the chamber. So the chamber will be used as a place to sterilize the equipment maybe with the ethylene oxide. This is an example of equipment that can be cleaned or sterilized with gas. Okay, so if you see over here, mostly they are made of plastic or rubber that you cannot use or you cannot put them on the heat type of sterilization. Aldehyde, like glutaraldehyde, they use as a sterilant. So this is a high level of chemicals. Orthotaldehyde, this is high level disinfection. It's not really sterilant, but at least it is a high level of disinfectants. For gas, again, the most common one is the ETO, the ethylene oxide. This is used as a sterilant for heat-sensitive equipment that made from plastic, for example. They usually sterilize with this gas. Detergent and soaps, mostly it's gonna be low level, right? Because it's gonna be used in our skin, in a living object, animate object. So it has to be low level of chemical. Otherwise, it might cause problem into our skin. So many of them is actually alkaline compound. Of course, they are because it is low level of chemicals. Agent is gonna be weak microbial. Sometimes it's kill only the least resistant type of microbes. It will not kill the intermediate and the high resistant type of microbes. This is just experiment showing us the difference between cleaning using the antiseptic soap with the normal non-antiseptic soap. This is the number of microbes in our hand, okay, after cleaning with the antiseptic and non-antiseptic soap. If you see over here, the concentration of bacteria stay the same. Maybe lowers a little bit when we use water to clean it, but after that it's growing back. But if we use the antiseptic, mostly it will reduce the number of microbes on our hand. This is example of some antiseptic and detergent, quartz, quaternary ammonium, and soap. Again, mostly they are antiseptic and disinfectant. So for this one is disinfectant. For the soap, it's gonna be antiseptic because it is used in the skin. Heavy metals, there are some heavy metal that can destroy bacteria or microbes. Example is silver, gold. They are also, okay, this is gold over here. They can also destroy bacteria. This is examples of experiment using silver and gold. So if you see over here, you have what we call the clear zone or zone of inhibition, which is no bacterial growth. So this is clear. Okay, no bacteria. So bacteria actually inhibited over here. So this is what we call the zone of inhibition. Okay. It means that bacteria is actually not growing because of this silver or gold. So these heavy metals kill and destroy bacteria that close to them. But if we see outside over here, this is bacteria that is growing. So you can compare from this experiment which one is more effective. So the higher or the wider the zone of inhibition, the more effective it's going to be. So if you compare between gold and silver based on this experiment, silver actually more effective compared to gold in destroying bacteria. Okay, another metal that can be used is mercury, for example. Silver, of course, silver. So silver has been used for a long time actually for killing or controlling bacterial growth. The last one is dyes. This is color, like paint, for example, that we use. They also have some ability of controlling or killing bacteria. Like some aniline dyes, for example, they are very active against gram-positive bacteria. Some even used as an antiseptic. So they are mostly low level and narrow spectrum. It means that just kill several type of bacteria, not many types. Okay, so acid and alkalis or alkaline. Some acetic acid can inhibit bacteria. Lactic acid can also kill bacteria. Therefore, they use for prevention of bacterial growth. So used as a preservative. Okay. Mostly they are low level type of chemical. So as you know, to preserve food, sometimes people put it in the acidic solution, or maybe in salty solution, or maybe in very sweet solutions, because this type of chemical can also prevent bacterial growth. Okay, I think that's all for chapter 11. So we finished chapter 11. We will continue with chapter 12 next. Goodbye now. Hey everyone, welcome to Medical Microbiology. Today we will discuss chapter 12, which is the drugs or chemotherapy, microbe and host, the elements of chemotherapy. There will be 5 lectures over this chapter. The first lecture will be about the introduction to the chemotherapy therapeutic agents, and also we're going to talk a little bit about their targets. Okay, so these are the terminologies of chemotherapy. So chemotherapeutic agent, okay, chemotherapeutic agents are any chemicals that use in the treatment, relief, or prophylaxis of disease. So it means that we use this chemotherapeutic agent or drugs, or sometimes we call also medicine, whenever the disease or the pathogens already inside our body. So this is used for inside our body. Okay, compared to chemical, physical agents that use for controlling the pathogenic bacteria or microbes outside our body. We already talked about that in chapter 11, which is prevention. This one is curation, which is we try to cure our body from disease that already inside our body. Okay, so this is the chemotherapeutic agents or drugs. And prophylaxis, this is a drugs that actually used as a prevention. Okay? So we take this drug, we take this medicine to prevent us against the disease. An easy example is Tamiflu. Okay, so Tamiflu is actually a drug that have both function. It can be used for curations of flu, okay, H1N1 if you have that flu, but it is also can be taken by the family member that do not have the flu, but try to protect from getting the flu from one of the family member that has flu. So this is what we call the prophylaxis, which is the medicine or drug that is used for prevention against some disease. Antimicrobial chemotherapy, this is a general term for any drugs that we use for microbe. Okay, so this is not for special type of disease. Okay, this is for the terminology for drugs that use for microbes. Okay, especially pathogenic microbes. And then so over here we have the antimicrobials again, which is the inclusive term for any antimicrobial drugs, okay, regardless of the origin. Okay, so this is, we use this as a general term for any drug that we take when we have the disease. Now for the antibiotics, this is a very specific term. Okay, this is only actually used for bacteria. So when we talk about antibiotic, it is actually only used for bacterial infections. So we cannot use antibiotics for curing the viral disease, okay, like flu, for example. So we do not take the antibiotics for flu because antibiotic is for bacteria. And the semi-synthetic drugs, this is combination between natural and made drugs in the lab. Okay, natural and the drug that is chemically modified in the lab, pharmaceutical lab. Synthetic drugs, this is the drug that modified in the laboratory, pharmaceutical factory or laboratory. Narrow spectrum drugs, this is used for the drugs or the medicines that is only used for specific infection. Okay, specific infection, for example, maybe only used for gram-positive bacteria. For the broad spectrum, it means that this is the drug that can be used for wide variety of infection. Okay, maybe gram-positive and also gram-negative bacteria. Now, this is very important, for using the drugs. The drugs should be used for destroying the infective agents or pathogenic microbes without harming, without harming the host cell or human cell. That's the primary principles of creating the drugs or using the drug for curations of disease or infectious disease. As it is, this is the ideal, this is the ideal characteristic of the antimicrobial drugs. First, which is it has to be, selectively toxic to the microbe only. Okay, it will not, it's not toxic to the host cell. It's not toxic to human cell. That's the primary or the first principle. The drug has to kill the microbes without killing our own cell. It has to be microbicidal. Remember, cide means kill. Okay, so the drug should kill the pathogenic bacteria or microbes rather than biostatic, rather only preventions of microbes, microbial reproduction. Because if it is microbiostatic, then this bacteria will be able to grow again, after several weeks of the drugs use. So it has to be the drug that able to kill or destroy the bacteria or the microbes. Relatively soluble because our body, is mostly consists of water. So these drugs should be soluble in our fluid, interstitial fluid or fluid inside our body. And then remain potent for long enough to act against that bacteria. It means that that drug will not be metabolized very fast in our body. Then it will lose its activity against the pathogenic microbes. Does not lead to development of antimicrobial resistance. So the drug that is used is not actually making the microbes become stronger. Okay, so we try to avoid this type of drug that causing the bacteria or the microbes to be stronger. And the drug should be complements. It is not the only chemicals that really kill the bacteria, but this is a complement or helper for our host defense. It means that our host defense still have to process the pathogens, the microbes, in addition to the drugs that being used. Remain active in the body tissue and the fluid, okay, readily delivered to the site of infection, reasonably priced, okay, does not disrupt the host health by causing allergy or this is what we call the side effects. Okay, so as much as possible, it has very low side effects to the host cell. So this is ideal. It means that sometimes it is very difficult to fulfill these requirements. For example, there are some drugs that is very effective, but actually that drug can also causing side effects to the host cell. Okay, so origin the antibiotics. So when we talk about antibiotics, this is only used for bacteria. Okay, so antibiotics, there are many of antibiotics actually made in nature. So this is a natural antibiotics. For example, they produced by some microorganism. For example, some bacteria is actually produce the antibiotics. For example, streptomyces, they produce, you know, a drug or antibiotic is called the streptomycin. Bacillus, maybe they produce other type of drug or antibiotic like bacitracin, for example. This is bacteria. Fungi or molds, in the genera of Penicillium, you know, it produce penicillin. Antibiotics called the penicillin. Cephalosporium, this is another fungi. So they produce antibiotics called the cephalosporin. So drugs, you know, the antibiotics can be produced in nature by some microorganism. So they actually produce this for compete with other bacteria or with other microorganism. Like penicillium, for example, it's produced penicillin in order to destroy some bacteria that may be around them. So this is kind of competition. Okay, so they produce to kill other microorganism, and then we actually harvest these antibiotics, and then we use in our body to kill the bacterial infection. And again, this is the primary principle for development of drugs or chemotherapeutic drug, which is it has to be selectively toxic. Okay, for the bacteria, maybe it's easier because bacteria cell is, you know, different than human cell. So the target that is used, you know, for the medicines is going to be easier to be used for bacteria, okay? Because they have different conditions, different type of cell compared to the human body. But it's going to be more difficult, okay, for infection that is caused by microorganism that is very similar to human cell. Like for example, if it is caused by fungi, okay, protozoa, worm, for example, it's going to be more difficult to find the selectively toxic drugs because why? Because these cells are similar, okay? So they are similar to human cell because they are eukaryotic cell, right? So it's going to be more difficult to develop drugs for this type of infections, okay, fungal infection, protozoal infection, and worm infection compared to bacteria over here. So that's the reason why you will find that there will be more antibiotics or drugs used for bacterial infection compared to the drugs that used for the fungi or protozoa or worm infection, okay? Because it's not easy to develop selectively toxic drugs if it is used for fungi, protozoa, or worm. So again, about narrow spectrum and broad spectrum. Narrow spectrum means a drug that only be used for certain type of infection, maybe for gram-positive bacterial infection only. But for the broad spectrum, this is a drug that can be used for many type of infection, including gram-positive and also gram-negative bacterial infection. Now the target, okay, remember when we discuss chapter 11, we also talk about the target for physical and mechanical agents on the microbes, okay? So it's going to be the same, it's going to be the same. So again, when we talk about bacteria have cell wall made of peptidoglycan, and then bacteria has cell membrane, okay, made of phospholipid bilayers, and bacteria has the DNA or chromosome, and bacteria also have the ribosome. Okay, so this is cell wall. Then under the cell wall, this is cell membrane. Okay, and this is DNA. Okay, and this is the ribosome. Okay, so remember ribosome is used for protein synthesis to make proteins, including enzyme. So bacteria are able to produce their own enzyme that is beneficial for themselves. Now, this structure will become the target for the drug. Okay? So this drug will have this target on this structure. Okay? So first target will be the cell wall, okay, inhibition of cell wall synthesis or destruction of the cell wall. Then the second target will be the cell membranes, okay, destruction of the cell membrane, for example. And the next target will be the DNA, okay, which is the nucleic acid synthesis, structure, and function of this DNA. Okay? And then the next target will be the inhibition of this process over here, okay, protein synthesis. So there are some drugs that will, destroy or block the protein synthesis inside the bacterial cell. And the last target will be the metabolic pathway. So these bacteria, they also perform metabolism. So there are some drugs that will block that process. the key process of metabolic pathway inside the bacterial cell. So these are become the target for drug. So there are, for example, some drugs that only target the cell wall. There are some drugs that only target the cell membrane, nucleic acid, protein synthesis, or maybe the metabolic pathway. We're going to see that type of drugs over here. So this is kind of summary and information about several drugs that is used on bacteria, and they have special target. Like for example, penicillin, cephalosporin, vancomycin, bacitracin, carbaphinams, fosfomycin, cycloserine, isoniazid. Okay? These drugs or medicine, they actually destroy this cell wall, right? So blocking the cell wall. So when the cell wall is broken, then this bacteria will be die. Okay, so this is the what we call the cell wall inhibitors. The second is going to be the cell membranes inhibitor or destroyers. Example is polymyxin. So polymyxin over here will destroy the cell membrane. The next drugs will be the one that target to the DNA or RNA. Okay, so this is DNA. So, you know, this bacteria have DNA, and DNA then will be able to produce the messenger RNA, and this messenger RNA then will be used for making protein by using the ribosomes. So there are some drugs that actually block this process. Okay, so protein, the nucleic acid synthesis. Examples is the quinones. So quinolones or quinone. Rifampin, for example, this is also used to block this DNA RNA synthesis. There is also drugs that has their target on the protein synthesis. Remember, protein is made by this ribosome, and the ribosome actually has two subunits. Okay, the large subunit and the small subunit. So there are specific drugs that only work on the large subunit like chloramphenicol, erythromycin, clindamycin, and oxazolidinones. These only blocking the protein synthesis on the subunit, the large subunit. There are also some drugs that actually work in the smaller subunit. So when the subunit, one of the subunit is blocked, then the protein, like enzyme, will not be produced, and then the bacteria will die because they need the enzyme in order to grow or in order to reproduce. Example of drugs that block the smaller subunit is aminoglycoside, tetracycline, streptomycin, and amikacin. Right? So these are the drugs that block the protein synthesis on the smaller subunit. And the last one will be the drugs that actually block the metabolic pathway. Right? So like, for example, bacteria able to produce a product from the substrate. So this require process over here. So there are some drugs that actually block this process. So when the process is blocked, then there will be no product. If there is no product, then the bacteria will die because they need this product in order to grow or in order to reproduce. Example of drugs that work in this blocking this metabolic pathway is the sulfa drug and trimethoprim. So this is just information about the drugs and the target, especially to bacteria. So we're talking about bacteria cells over here. What are the target for each specific drugs? Okay. Now, as we mentioned before, that the drugs that is used for bacteria usually have more diverse type, because bacteria cells are different than human cell. So it's gonna be easier to make the drugs that selectively toxic to bacteria only, without causing problem to the host cell. Right? Because what? Because bacterial cell are different than human cell or the host cell. So if you see over here, there are so many drugs that actually used for bacterial infections. Okay? And then from here, you can also what is the target for each of these drugs, like penicillin, cephalosporin, bacitracin, aminoglycoside, for example. The bacitracin over here, they all have the same target, which is the cell wall. Aminoglycoside, they use for RNA, protein synthesis. And then erythromycin, for example, the same, is blocking the protein synthesis. Vancomycin, also cell wall, the same like the penicillin. Tetracycline is gonna be protein synthesis. Chloramphenicol is the same, but remember they have different site. One is in the larger subunit, the other one is the smaller subunit of ribosome. Quinones, this is blocking the DNA and RNA synthesis. Rifampin, also RNA synthesis. Sulfur drug, this is the one that block the metabolic pathway. The same with the trimethoprim. So again, you will find more drug that used for bacterial infection compared to the infection that is caused by fungi, protozoa, or worms. Over here, including viruses. Why? Because these fungi, protozoa, and worms or helminths, their cell are very similar with the human cell. So this is a eukaryotic cell, right? So eukaryotic cell, so they are similar. Therefore, if we use the drugs targeting this cell, it might also targeting our own cell. So therefore, again, selectively toxic drugs that is made for this type of infection usually more difficult to make compared to drugs for bacterial infection. So you see less drug is used for fungi, protozoa, or helminth. Fungi, for example, amphotericins. This is for cell membranes. Azoles for also cell membrane. Flucitocin. This is for inhibiting the RNA synthesis. For protozoa, like malaria, for example, quinones, or metronidazoles, helminths, bendazole diethylcarbamide, piperazine, niclosamide, ivermectin. This is for helminths or worm infection. Now, how about viruses? Viruses is not similar with our cell, right? So even viruses is actually not a cell. Why? There are less drug for viral infection compared to bacterial infection. The reason is because virus, when it is infecting our cell, it will enter into our cell. Compared to bacteria, usually they are outside the cell. So virus, they will enter into our cell. Remember the viral cycles? There are 6 cycles. Start with penetration. The attachment, penetration, uncoating, synthesis. After synthesis, there will be assembly and then release. So this process is happening inside the cell. So therefore, if we want to make a drug that kill this virus, most of the time this drug will also causing problem to the infected cell, to our own cell. So there will be more difficult to find the drugs for virus that is selectively toxic only to the virus without causing problem to our cell. Example for this viral infection or drug is the oseltamivir, cyclovir. This is mostly for herpes virus, for example. This is azithromycin or AZT. This is mostly for HIV infection. Okay, it's also known as AZT. So this is examples of drugs that's used for different type of infection. I think that's all for lecture number 1. So in lecture number 2, we will visit again the drugs and the target closely. So we're gonna see which drug that actually destroy the cell wall, destroy the cell membranes, protein synthesis, DNA, and block the metabolic pathway. I think I see you on the next lecture. Goodbye now. Hi everyone, welcome to Medical Microbiology. So today we will continue chapter 12, chemotherapeutic drugs. And today we are in lecture number 2, which is we will talk a little bit about the drugs and their targets. So remember this picture, this diagram. Again, if we talk about the bacterial cell, it has cell wall, cell membranes, DNA, which is chromosome, of course, ribosome for protein synthesis, and some metabolic pathway inside the bacterial cell. So this is, we're talking about process or structure of bacterial cell, which is our target for taking the drugs or the antibiotics. So there are some drugs that will inhibit the cell wall. There are some drugs that will inhibit the cell membranes. There are some drugs that used for blocking DNA or RNA replication. There are some drugs that's used for blocking the ribosomes. And then there are some drugs that's used for blocking the metabolic pathway inside the bacterial cell. So if this structure and also process being blocked, then the bacterial cell eventually will die. Okay, so now we start with the one that block the cell wall, okay, bacterial cell wall. So remember, bacterial cell wall made of peptidoglycan. So this become the target for the drug, which is destroying the peptidoglycan. Examples of that drug is in the class of penicillins, and mostly this one will be very effective for gram-positive bacteria. Okay, remember gram-positive bacteria, they have thicker peptidoglycan. Therefore, this peptidoglycan will be easier to be destroyed by the penicillin. Compared to the gram-negative bacteria, they have outer membranes that kind of protect the peptidoglycan from the drugs. So usually, the drugs that effective against the cell wall mostly used for gram-positive bacteria, Like penicillin, for example, is used for Staphylococcus, Streptococcus, which is gram-positive bacteria. And they usually not used for like E. coli infection because E. coli is gram-negative bacteria. But there are also some broad spectrum of penicillin that can be used for both gram-positive and gram-negative bacteria. But just remember, penicillin mostly more effective for gram-positive bacteria. So this is really important when you do the experiment, identifying whether the bacteria is gram-positive or gram-negative. It is also used for giving the correct drugs for treatment of this infection. So you, for example, the doctor will not give prescription for gram-positive bacteria infection if the infection is actually caused by the gram-negative bacteria. Okay, this is what happens when the cell walls are broken. So like penicillin will break the cell wall. Then what happened? The water will enter inside the bacterial cell, and bacterial cell is become bigger, and finally, it will explode. This is from the electron microscope. So what happened with this bacteria that the cell walls has been broken is become bigger because of water is actually enter into their cell, and some of them is already explode. So they die because their cell become explode due to the entrance of water into their broken cell. Now the one that destroy the cell membranes, the example is polymyxin. So this polymyxin is actually separate the lipids, the phospholipid bilayer, and causing also leakage to the cell. Okay, so this one usually used for gram-negative bacteria. So this polymyxin is actually break this cell membrane. The drug that inhibits nucleic acid synthesis, example is chloroquinone and quinones. Some of them, for example, chloroquinone is actually bind to the double helix DNA, so the DNA will not be functional. And quinone, for example, they actually inhibit the helicases. Remember, when the DNA start to replicate or duplicate, they need to be unwind because this is double helix structure. Okay, so in order to be replicate or duplicate, this double helix has to be open. So like to be open like that. So therefore, the new DNA will be made over here. Okay. Now, in order to open this double helix, they need enzyme is called the helicases, and quinone is actually block this enzyme. Therefore, the bacteria DNA will not be able to replicate. The drug that block the protein synthesis, okay, several of them, like for example, aminoglycoside will block the 3S subunit, which is smaller subunit. So if you remember this is bigger subunit, and this is smaller subunit. So this is 50S, and this is the 30S unit. So aminoglycoside is actually block this smaller subunit. So tetracycline, for example, will block the larger subunit. Okay, so this is example. This is aminoglycoside. Is actually here. Okay, so they attach to the smaller subunit, causing the messenger RNA will not be able to convert it or translate it into the protein. So there is no protein production over here. For chloramphenicol, for example, this is on the blocking the larger subunit. So when it is blocked, then messenger RNA will not be able to translate it into the protein. Other like chloramphenicol, tetracycline, erythromycins, they are blocking between this bigger and smaller subunits. So the process will be blocked over here. So no protein production. And the last one is drug that affect the metabolic pathway. Example is sulfa drugs and rifampin. So sometimes they do this process by competitions with the process, with the substrate, for example. Okay, this is examples. The use of sulfa drugs and trimethorphan. So sulfa drug usually used for the first process over here, which is competition with the substrate. Okay, so this is the normal process for bacteria to produce the product over here. So this product is amino acid that is important for them. So they need this amino acid in order to perform their process, maybe growing or reproduction. So the normal process is they have the substrate called the PABA or PABA, and this PABA will enter or will combine with the enzyme, okay, and then they will produce the intermediate products over here. And then after that, the intermediate products will be used to make these final products. Okay, so this is the normal process. So when they able to produce this, then they will growing very fast, they reproduce and etc. Okay, so the drugs, sulfa drug, for example, is used to block this process over here, compete with the substrate. So this is substrate is look like, okay. Right, so this is the normal process. The substrate will bind to the enzyme. So this is enzyme, and finally it will produce the intermediate product, okay, which is this one. Okay, so the process then continue to produce the final products over here, the amino acid. Now, when the sulfur drug coming, so this is sulfur drug. If you see the sulfur drug is very similar with the substrate over here. It's kind of similar, right? So that's the reason why, you know, this sulfa drug actually compete with the substrate to bind to the enzyme. Okay? So when the sulfa drug that actually bind to the enzyme, then there will be no intermediate product of here. So naturally, you know, normally, you know, the substrate will enter into the enzyme and produce this. However, because the sulfa drug enter into this combination, okay, so enzyme and sulfa drugs complex, then there will be no production of intermediate product. So if there is no intermediate product, then there will be no final product. So that's the sulfa drug. The trimethorphan, okay, is worked on the second process over here. So let's say there is no sulfa drug, so the bacteria able to produce the intermediate products over here. Then what happens? The trimethorphan enter into this metabolic pathway and block this process. So if the process is blocked, then there will be no amino acid production. So this is how sulfa drugs and trimethorphan block the metabolic pathway. They are actually blocking different side, not the same side of pathway. Yeah, I think that's enough for lecture number 2. We will have a next lecture, especially we're gonna talk about the antibiotics, which is the drugs that used for bacterial infection. Okay, I see you later. Goodbye now. Hi everyone, welcome to Medical Microbiology. So today we will continue chapter 12. We will discuss about the antibiotics, which is drugs that use for bacterial infection. Okay, so this is type of antibacterial. Okay, just remember, okay, so what we call the antimicrobial drugs. So this is a general term for drug that used for any type of microbial infection. It can be divided into several groups. The drug that only used for bacteria is called the antibacterial drugs. Okay? So this is antibiotics. So remember, antibiotic is for bacterial infection. If it is natural, production is produced by nature, then it's just called antibiotics. But if it is made in the laboratory, then this antibacterial drug is called as a synthetic drugs. Okay? And this is the one that we will discuss in this lecture, okay, lecture number 3. And then if it is used for fungi, then it is called the antifungal drugs. If it is used for protozoa, then it is called the antiprotozoal drugs. If it is used for virus, then it is called the antiviral drugs. This is actually one more, but it will not be discussed in chapter 12, which is anti-helminthics, which is worms. Okay, anti-helminthic drugs, which is for worms infection. Okay, so in this lecture, lecture number 3, we will discuss these antibacterial drugs. Then we will discuss these 3 types of antimicrobial drugs in lecture number 4. Okay, so antibacterial drugs or antibiotics. The first one developed is actually the penicillin. And then after that, the drugs or the antibiotics are becoming more and more and more. There are some natural products, and there are also some drugs or antibiotics is produced in the pharmaceutical lab. So this penicillin is actually produced naturally by fungi, which is in the species of Penicillium chrysogenum. So this is the major source for natural penicillin. So this is the list of the penicillin drugs from the first type, which is natural type, penicillin G. So this is naturally made by Penicillium fungi, and then penicillin V. So this the natural type. It is made by Penicillium. And then after that, some drug is become semi-synthetic. Some is actually produced in nature, and then there are some modification in the lab. It's become semi-synthetic drug. And finally, the last generation, there are some drug that especially made in the lab, which is synthetic type of penicillin or antibiotics. Okay, so these drugs, the one that destroy the cell wall, mostly they have the same nucleus structure. or center structure. It's called a beta-lactam structure. So all the drugs that used for destroying bacterial cell wall, they usually have this beta-lactam structure. That's the main structure for this type of antibiotics or drugs. So this is the nucleus of the main structure over here. Okay, so the beta-lactam structure. The difference between the drugs, different between the penicillin, cephalosporin, amoxicillin, for example, usually on this, what we call the R group or side group or radical group. So this make different type of antibiotics, different type of effectiveness, whether broad spectrum or narrow spectrum, and etc. So the pharmaceutical company usually playing in this area over here on the radical group. So they change the radical groups in order to produce maybe more effective or broad spectrum type of antibiotics. Again, penicillin G and V, this is the most important natural form. So this is made in nature. And then there are development of semi-synthetic penicillin, which is some part is made by nature, and then in the lab they put some additional structure, especially radical structure, in order to produce more broad spectrum drug. And then there are also some antibiotics that all the process is made in the lab, which is the synthetic drugs. The same thing with the cephalosporins. So initially it is made in nature by this fungi, okay, Cephalosporium acromonium. So this is fungi that produce this cephalosporin. And then the first generation usually natural made cephalosporin. Then in the lab, they're creating another generation, like second generation, third generation, and fourth generation that is created in the lab. And this new generation usually more broad spectrum type, so widest range of use for different type of infection. Again, the way they made that second, third, and fourth generation is just playing with the R group over here, radical group, which is radical group number 1 or radical group number 2 or side group number 2. The nucleus or the center is gonna be the same. They all have the beta-lactam structure over here. So this is beta-lactam structure. Okay, so they just modify these radical groups in the lab, in order to create a new generation of cephalosporin that is better for curing wide type of infection, maybe gram-positive and also gram-negative bacterial infection. Additional beta-lactam drug like carbapenems, monobactam, this is also used for mostly for gram-positive bacteria, because gram-positive bacteria, they have the thicker cell wall that usually is become the target for these beta-lactam drugs. And again, remember, gram-negative bacteria, their cell wall is actually covered by additional membrane, which is a protein or lipoprotein membrane that sometimes protect them from this type of cell wall destroyer. So usually the one that is used for cell wall destruction, mostly more effective against gram-positive bacteria. But again, the broad spectrum type of penicillin, cephalosporin, they also able to destroy the cell wall of gram-negative bacteria. There are also some non-beta-lactam cell wall inhibitors. Okay? So like, for example, bacitracin. These drugs or these antibiotics do not have the beta-lactam structure, but they also able to destroy the cell wall. Mostly, this bacitracin is used as a topical agent or topical medicine or ointment. There are another one is isoniazid or INH, mostly used for bacteria that actually cannot be classified as a gram bacteria, whether positive or negative, like Mycobacterium tuberculosis. Remember this Mycobacterium TB, it has cell wall, but the cell walls, cannot be really tell whether it's special cell wall is different than cell wall of gram-positive bacteria and gram-negative bacteria. So it has different type of cell wall. I think that's all for antibacterial drugs. And next lecture, lecture number 4, we'll talk about the antifungal, antiparasitic, which is antiprotozoal, and antiviral drugs. See you on the next lecture. Bye. Hi, everyone. Welcome to Medical Microbiology. Today we will continue chapter 12. This is going to be lecture 4. We will talk about the antifungal, antiparasitic, and antiviral drugs. So remember again, there are several groups of antimicrobial drugs. The first one is used only for bacteria, therefore it's called the antibacterial drugs. We already discussed this on the previous lecture. And then if it is used for fungi, it's called the antifungal drugs. If it is used for antiprotozoa, for the parasite protozoa, for example, it is going to be antiprotozoal drug. And if it is used for virus, then it's called the antiviral drugs. This is again one more is anti-helminthics. If it is used for worms infection. Okay, just for information, number 3 and 5, they actually can be combined into what we call the anti-parasitic drugs. Okay, so antiprotozoa and anti-helminthics is considered as the anti-parasitic drugs. We will also talk about this in today's lecture. First one, okay, it is actually the second one. Remember the first one we already discussed on the previous lecture, which is antibacterial drug. Now is the antifungal drugs, okay, the one that's used for fungal infection. There are several of them. The most common one is the amphotericin. Okay, this is used for fungal infection. Nystatin is also for fungal infection, especially for topical treatment. For the stubborn type of fungal infection, like athlete foot, for example, greaseylfulvin usually used for that treatment. Azoles, okay, this is for broad spectrum of fungal infection. Flucytosine, this is also for fungal infection, and echinocandins, also used for fungal infection. This is macrolide, a type of drugs that also used for treatment of fungal infection. And the next one will be the antiparasitics. Remember, these antiparasitics will be 2 types. The first one will be protozoa. Okay, anti-protozoal drugs. And the second will be anti-helminthics drugs, right, which is used for worm infection. Okay, so anti-malarial, for example, so malaria is actually caused by protozoa. So this is going to be anti-protozoal drugs. So like quinine, chloroquine, this is the main drug that's usually used for malarial infection. And this drug is also used for prophylaxis. Okay, remember again the terminology, lecture 1, chapter 12. We also talk about the terminology for prophylaxis, which is drugs that also used for prevention against disease. So quinine, for example, in some area is also used for prevention. So for example, if someone want to go for going to the area that a lot of malaria infection, then they can take this quinine as a prevention. Another anti-protozoan drug like metronidazole, this is also used. Remember this tetracycline, this is actually antibiotics that also actually can be used for anti-protozoal protozoal infection. For the anti-helminthics drugs, so some of them, mebendazole, thiabendazole, niclosamide. So this is also used for worm infection. The next one is the one that used for viral infection. It's called the antiviral drugs. There are several types of them, like acyclovir. So if you see this acyclovir, mostly for herpes viruses. Ribavirin, usually for some hemorrhagic viral infection. AZT, this is usually used for HIV infection. Now, how this antiviral drugs actually used for? So what kind of targets? So the target mostly on the, related to the host cell. Okay, so this is, let's say this is the host cell. Okay, so there are some drugs that use for blocking the attachment and also the penetrations of the virus into the host cell. So remember again, there are 6 stages of viral cycles. Right? So start with the attachment, penetration, uncoating, synthesis, assembly, and the last one is release. So when you remember that each process can be a target for the antiviral drugs. So therefore, the first group, gonna be the one that block the attachment and penetration, for example. Like for example, fusion, this is used for blocking the attachment of HIV viruses. And amantadine, this is used for blocking the flu virus attachment. Right? The same thing also, Tamiflu is also used for blocking the penetrations of flu viruses. So this is a group of drugs that use to block the penetration and also attachment. Okay, Tamiflu and amantadine also used for blocking the release of the the virus, right, from the host cell. Now, the next group will be the one that inhibits the nucleic acid synthesis. Okay, remember bacteria carry nucleic acid, maybe DNA or RNA. Inside the host cell, this DNA or RNA virus will be able to duplicate or replicate. So there are some drugs that will be targeting this, targeting this process, whether duplication, replication, transcriptions of this nucleic acid. Okay, example is acyclovir. This is for herpes. So it will, block the synthesis of DNA of these herpes viruses. AZT, okay, this is also will block the synthesis of DNA and RNA of these HIV viruses. Remember, HIV is actually RNA viruses that able to replicate into DNA, and then it will enter into the host cell to combine with the DNA, the host cell DNA. Okay? So AZT actually block this process. So there are some other drugs that also able to block the process of replication and transcription of these nucleic acids of some viruses. The last one is the one that inhibit the assembly and also release of the virus. So maybe the virus is still able to produce the protein like capsid, the spike, and etc. But this drug, is used to block the combinations or the assembly of this capsid and the DNA. So basically, the virus that will be produced because of this blocking will be imperfect type of viruses, is not complete viruses. So it will not be able to causing infection to the other cell. Or maybe some drugs that will block the release. So the virus basically just stay in one cell, unable to exit the host cell to infect the other cell. So this is a group of antiviral drugs. Again, there are three groups. The first one usually used for blocking the attachment and the entry of the virus into the host cell. Okay, that's the first group. And the second group will be the drugs that use for blocking the synthesis of the viral cell. Right? And the last one will be the drug that can block the release and also the assembly of the virus inside the host cell. Okay. Again, this is example of some antiviral drug, especially the one that used for influenza, amantadine, rimantadine, Relenza, and Tamiflu. And again, I mentioned before that Tamiflu is not only used for curing the flu, but also can be used as prophylaxis or preventions for flu, especially H1N1 type of flu or influenza A type of infection. Another example is anti-herpes drugs. So mostly is end with fir, like acyclovir, valacyclovir, famiciclovir, pheniciclovir. So all of them used for herpes, whether labial herpes or the genital herpes. Remember, labial herpes is caused by herpes simplex virus number 1. The genital herpes is caused by the herpes simplex virus number 2. So these are different type of anti-herpes drugs. For HIV, there are several type of drugs that is used. Okay? Like the most common one is AZT over here. This is the one that block the synthesis of DNA or DNA replications of RNA replications of these HIV viruses. There is another drugs or pharmaceuticals medicine that also used for infectious disease to cure the infectious disease. This is called the interferon. So interferon is actually produced by cell, the infected cell. Okay, so let's say this is a cell. This is the host cell. Okay, so a human cell, for example. If there is a virus enter into the cell, or maybe some infection into the cell, so this is considered as infected cell. So this cell is actually able to produce the interferon. So this interferon then will be used by the healthy cell. Let's say this is another cell. Okay, healthy cell. So when the healthy cell receives the interferon, this healthy cell will be able to produce the protein that will block the entrance of the virus, for example. So this interferon is actually used for making this block proteins, okay, the protein that able to block the infection. So this interferon actually can be harvested. Okay, so when it is harvested, then it can be used for disease to cure the disease. Okay? Like for example, is used as a injection. So it injected into the healthy human in order to prevent them against some disease. Example is if a doctor or a nurse taking care of hepatitis B patient, sometimes they get the interferon injection in order to prevent them from getting the hepatitis B infection. Okay, this is just for examples. Some other also used for example, treatment again, hepatitis C, genital wart, kappa C, sarcoma, and etc. So there are many functions of these interferons. I think that's all for lecture number 4. We will continue with lecture number 5, which is special discussion on drug resistance and also drug tests, looking for MIC, minimum inhibition concentration, and TI, which is therapeutic index. So I will see you on the next lecture. Goodbye. Hi everyone, welcome to Medical Microbiology. So today we will continue chapter 12. This is going to be the last lecture, which is lecture number 5. We will talk about the drug resistance bacteria. We're going to talk about the tests for effectiveness of the drugs, and also we will discuss a little bit about MIC, minimum inhibition concentrations, and TI, therapeutic index of drugs. So we start with the drugs resistance. Okay, so remember the ideal drugs should be the drugs that will not will not cause the bacteria to be resistant to the drugs. Okay, so the bacteria that resistant to drug is mean that that bacteria is actually stronger, and then in order to kill them, then we have to increase the dose of the drugs. Okay? So now the question is how these bacteria are able to be resistant. We're gonna see a little bit about how they actually have the resistant gene. Again, remember when we talk about the structure of bacteria, okay, in chapter 4 and 5, okay, when we talk about the prokaryotic cell, remember there are some bacteria that actually have the pilus, or pili, which is the channel that can be used to transfer gene to other bacteria. Okay, so this is the what we call the pilus or pili, which is the channel used to transfer something from one bacteria to another bacteria. So many gram-negative bacteria like E. coli, Pseudomonas, for example, they have the ability to transfer, let's say this is a gene, special gene is called the resistant gene. Okay, this resistant gene actually can be transferred into another bacteria over here. And what happened, this bacteria then is become resistant. Okay, so this is a natural ability of some bacteria that are able to transfer resistant genes. So E. coli, for example, they're able to transfer the resistant gene to another gram-negative bacteria like Bacteroides, for example, Neisseria, Shigella, Klebsiella, so Serratia, for example, able to transfer the gene to E. coli. E. coli able to transfer the gene to the Pseudomonas aeruginosa, or Pseudomonas able to transfer the gene to E. coli. So you see this one, okay? So this is how they able to transfer the resistant gene to many bacteria. They're also able to transfer cross groups. So this group over here is actually gram positive. Okay, so this group over here is gram negative bacteria. So mostly gram negative bacteria are able to transfer the gene between them. And they're also able to transfer the gene to the gram positive bacteria, like gram positive bacteria. For example, E. coli, they're able to transfer to Bacillus, or Staphylococcus aureus. This is gram positive to another gram positive bacteria. So this is a natural kind of process that some bacteria have the ability to transfer their resistant gene, sharing their resistant gene to another bacteria. Now, the mechanism that they perform, so if that bacteria, for example, have the resistant genes, they usually have the ability to perform this process, okay? For example, Staphylococcus aureus, they have the ability to inactivate the drugs, penicillin. So inactivate the drug. So they have, or they produce an enzyme is called penicillinase. So this resistant gene produce the penicillinase, and this penicillinase is actually will destroy or block the penicillin. So when we use penicillin to this bacteria, then it will not be effective because it will be broken down by the penicillinase. So example of bacteria that produce penicillinase is Staphylococcus aureus. So Staph aureus, okay, this bacteria actually have the resistances against penicillin. So it's mostly penicillin will be not effective against Staphylococcus aureus. So this is the first process that may be happening in bacteria that have resistance against drugs. Another process may be by decreasing the permeability of the drug. It means that some bacteria that have resistant gene, they're able to produce proteins that will block the entrance of the drugs. So, for example, if we take the drugs, then this drug will not enter into the bacterial cell because of this action. Okay? The next action will be increase the eliminations of drugs from the cell. Okay? So again, some bacteria that have resistant gene, the drugs will be enter into their cell. Okay, so let's say this is a drug enter into the cell, but this bacteria is actually have the ability to pump this drug out from the cell. Right? So increase the elimination of this drug from the cell. So therefore, these drugs will not be effective against this type of bacteria that have the ability to remove the drug from the cell. Or there are some bacteria that have the ability to produce receptors that will not recognize the drugs. So for example, let's say this is a bacterial cell. Usually there is a receptor that will receive the drugs. So if this bacteria have this receptor, then this bacteria will be mostly killed by the drugs. Okay, but there are some bacteria that produce another type of receptor that will not bind the drugs because different shape, for example, like this. This one is curved. It will be. So when this bacteria have the ability to produce this receptor type, then the drugs will not be able to bind to the receptor, and this bacteria will be able to protect themselves against the drugs. Okay, so they change the receptor. So if they use to have this kind of receptor, then they change it. They make another type of receptor that is safe from binding the drugs. And the last action will be, there are some bacteria, if they do have the resistant gene, they're able to change their metabolic patterns. Remember, sulfa drugs, they're able to compete with the substrate in order to block the metabolic pathway. So if the bacteria has the resistant gene, they able to change that pathway. Therefore, sulfa drug, for example, will not be effective against this type of bacteria that able to change their metabolic pathway. Okay, we're gonna see the picture of that activity. Again, the first action may be drugs inactivation. So this is penicillin, like this. So if bacteria are able to produce penicillinase, this penicillinase actually will break this penicillin into a type that is inactive against the bacteria. So bacteria will be resistant against penicillin because it produces penicillinase that break the penicillin. The second action again is to decrease the permeability. It means that the drugs will not be able to enter into the cell. Right? So this one is the drugs able enter into the cell, but because it's this bacteria able to decrease the permeability of the drugs, that most drug will be outside the cell and this bacteria will be safe. And the next one will be the again the activations of pumping the drug out, removal of drug from the cell. So let's say the drug is actually enter into the cell. However, this cell, the one that is resistant, has the special pump over here that will pump the drug out. So finally, the drug actually will not stay in the in the bacterial cell. So this bacteria become resistant against this drug. The next action is by changing the binding site. So just remember, for example, there are some drugs that bind into the smaller subunit or bigger subunit over here. So if the bacteria that have resistant gene, they're able to change this receptor. So these drugs over here is actually will not be able to bind to the bacteria that able to change the receptor on the ribosome. So this drug actually will not bind to the ribosome. So, you know, the bacteria will be protected. And the last one is the bacteria that have the ability to change their metabolic pathway. For example, this is the normal metabolic pathway, and then if you put the sulfate drugs over here, then there will be no product. Okay. Now, this bacteria has the ability to change this pathway. Instead of from B into C, they change it into B into C1. And when they change the metabolic pathway from B to C1, these drugs will not be effective against this new metabolic pathway. So basically, then the bacteria will be safe. They will be able to produce their main products. So these are the mechanism of resistance, okay, bacterial resistance against drugs. There is another resistance with the process what we call natural selection. Okay, so just remember, in one populations, let's say human populations, we have mostly a regular type of people, right? But there are also some people that is actually not a very they have special condition. Maybe they are stronger compared to just regular people. So the same thing with this bacteria in population, mostly will be just regular type of bacteria, but there are some of bacteria that's actually really strong. Okay, so this bacteria is actually resistant against drugs. Okay, let's say we put drugs into this population, boom, and then what happened? Mostly the regular bacteria will be killed, and then only the strongest bacteria stay. And then after several weeks or months, for example, the one that develop inside the cell will be mostly the resistant bacteria. Okay, and then this will be growing as a new population over here. And these resistant bacteria, actually, there are some of them will be also even stronger. Okay, so this one is become regular now. So in order to kill this stronger bacteria over here, resistant bacteria, then we need to increase the dose, so higher dose of drugs. And this higher dose of drugs will mostly kill the resistant bacteria over here, but not killing the very resistant bacteria. And what happened then, this bacteria is the one that will become the new population. And in order to kill this new population, we need the even higher dose of infection. So this is what we call the natural selection of drug resistance in bacterial population. Now, this is a very important for us and also for physicians and government. So when we use antibiotics or drugs, so physician has to be sure that they're giving the right prescription, the right drug, the correct drug that will kill or cure the infection. So the physician will not give the drugs, kind of trials drugs to the patient. Otherwise, it will cause the infection become stronger, or the bacteria become resistant. And for us as a patient, this is really important that we have to comply with the rule. So like, for example, if a doctor telling us to finish the drug within 10 days, then finish it in 10 days. So do not stop taking that drug. For example, maybe in the 5th day, we already feel better. We think that we are better. It is actually not, because not all bacteria actually kill in 5 days. There are still some strong bacteria still alive. So if we stop taking the drugs in the 5th day, then what happens? The strong population will be growing inside our cell. And if we need to kill them later, then we need higher dose. So if the doctor say finish the drug within 10 days, then finish it, all the drugs that doctor give to us. Otherwise, if we just use half of them, then there will be a resistance. The strong bacteria will still be reproduce or replicate inside our cell. So administrations drugs, okay, is also to be careful. So there are some drugs is broad spectrum or narrow spectrum. If the doctor or the pharmacy already know that the infection is really special or specific, it's only one bacteria that causes infection, it is better to just give the narrow spectrum of drug that only kill that bacteria. Because if we use the broad spectrum, it might kill other bacteria that may be efficient for us, benefit to us. So it's really important, again, the drug administration, to give the correct drug. The best drug will be the narrow spectrum if the infection is only one type of infection. If it is mixed infection, then it can be given the broad spectrum of drug. So for the pharmaceutical company, so drug research, for example, they need to focus with the drug that have a short-term effect. It means that it's not causing the resistance to the drugs, because it's long-term use. It might increase the capability of the bacteria to be resistant against that long-term type of antibiotics or drugs. And then long-term strategy, this is mostly for government. For example, there should be rule of using the drugs, the antibiotics. Sometimes farmers, for example, they also use the antibiotics to their cattle. So if they do not use correctly, so the bacteria inside the cattle is actually become stronger and stronger, and it can jump to give the infection to human, for example. And this, the one that give infection to human, will be stronger type of bacteria, because they already resistant against the drugs that is given to the cattle, for example. Okay. There are some people that actually have a side effects against the drugs. So, but 5% mostly drugs can cause side effects, especially to some people that have kind of lower immunity, or they have some problem with their body. They might develop the side effects. For example, maybe the side effect could be the direct damage to the tissue due to the toxicity of the drugs. So like, for example, HIV drug usually causing this problem to the user. Okay. Allergic reaction. Some people have allergic reaction against penicillin, for example, or maybe disruption of balance of the normal flora. And when it is happened, that is can cause what we call the super infection. So what is the example of this super infection? Let's say we have the infection in our throat. Okay, so we give the drug. Let's say we give vancomycin. Okay, vancomycin usually used for throat, like strep throat infection. So this vancomycin actually will kill this bacteria over here, but it also going down to the GI system. And finally here in the intestine, these drugs able to kill the natural flora. So indigenous flora or natural flora that is important for us. This is what we call the probiotics, okay? So this is the one that important for us. This natural flora actually compete against the pathogenic flora over here. Okay? This pathogenic flora will not cause infection to us because they compete with this natural flora. So this natural flora actually keep this pathogenic flora in balance. So when this drug enter into this system, what happened? It kill the natural flora because maybe natural flora is more sensitive against these antibiotics. And what happened? This one is growing. And finally, the one that's growing in the intestine is pathogenic flora, and then it causes the disease. So this is what we call the super infection, which is the infection in the site due to the use of antibiotics on another site. So the infection that's happening in this intestine is called a super infection. This is the idea why people, for example, suggest to take the probiotic in order to grow more natural flora. You can read this in your textbooks. This is some major side effects on human body. Okay, like for example, penicillin might have a side effect on the skin, like rash, itchy. Tetracycline, if I hear, for example, it might have effect on discolorations of teeth. So the teeth become yellow for longer use of tetracycline, for example. So this is again some list of side effects on our body caused by the antibiotics. Okay, this is consideration of selecting the antimicrobial drugs. So usually there is a test to know whether the drug is effective or not, and what is the best concentrations of the drugs. Okay, what is the best dose for the drugs in order to cure the infection. There are several types, okay, of identification. Okay, like for samples, to test whether the drug is effective or not, usually using what we call the Kirby-Bauer division test. Okay, so this is usually for effectiveness. Effectiveness of drugs. MIC, for example, over here, this is usually to know the dose of drugs. Okay, so what is the best dose used for making the drugs that will kill or destroy the infection? Okay, this is the Kirby-Bauer test, which is the division test in order to test the susceptibility or sensitivity of drugs against bacteria. So this different color over here is actually, this is bacteria growth. Okay, this is bacteria growth. Okay, so when we put the drugs or antibiotics on this bacteria population, like say for example here, Okay, chloramphenicals, this is ampicillin, okay, gentamicin, for example, enrofloxacin, oxytetracycline, cefotaxin. Okay, so 1, 2, 3, 4, 5, 6. So 6 different drugs is actually used for this test. Okay, let's see what type of bacteria they use. It doesn't say, but let's say this is used on Staph aureus, Staphylococcus aureus. Okay, so this is actually a lot of Staphylococcus aureus growing in this petri dish, right? So when we put the drugs in it, and what happened, there is a zone of inhibition over here. Okay, so this is a clear zone, okay, or zone of inhibition. Okay. What does this mean? In this clear zone over here, okay, there are no bacterial growth. Because of what? Because this drug actually kill bacteria in this area. So you see this one, this area is clear, this area is clear, clear area, right? This clear area is mean that there is no bacterial growth. So we can measure this clear area, you know, by using ruler, for example. So the wider the area is mean that the more effective the drug's gonna be. Like for example, this enrofloxacin actually the most effective drugs against this. Let's say this is Staphylococcus aureus because it has wider zone of inhibition around 10, 20, 25, 26, 27, okay, 27 millimeter wide. Is really effective compared to this one, for example. So oxytetracycline is only maybe what, maybe 10 millimeter. So this is the least effective one. This one may, you know, the next one that is effective because it is also wide. This one may be the third effective. This is maybe the fourth effective. It's almost similar. This is the fifth effective one, and this is the least effective type of drugs. So this is what we call the Kirby-Bauer diffusion test to test the effectiveness of antibiotics against certain type of bacteria. So we can change this bacteria. Maybe we use E. coli, for example, or we can use another type of bacteria to check to know which antibiotic is actually more effective against this type of bacteria. So based on this, then, you know, when the doctor give the prescription, then the doctor will give this one over here because this is the most effective one. The doctor will not give this one because this is very less effective. Okay, this is the test for identifying the dose that is given to the drug. It's called the MIC. Okay, MIC, which is minimum concentrations, minimum inhibitions concentrations. Okay, there's the lowest concentration that inhibits bacterial growth. So minimum inhibition concentration. Now, how the test done? This is by, so this tube is actually have bacteria on it. So before this tube given the drugs, okay, so the drugs will be put into this tube. So this tube actually has bacterial growth, like the control over here. So all of them start with this, this color, all of them. Now, the control, we will not put any drugs in the control. So zero concentration. On the first tube, Okay, after the control, then we put, let's say, 0.2 dose milligram per liter, for example. And then on the next tube, we double the concentration, 0.4. We put it over here. And then we double 0.8, double 1.6, double 3.2, double concentration 6.4, double the concentration 12.8. If we have another tube over here, then it's going to be around 26 point, 25.6 dose. Okay. So we put that drugs with this concentration. What happened after several days, maybe 1 day or 2 days, we check what happened. So basically in the control, because there is no drugs, the bacteria will grow. So you will see this bacteria over here, okay? So cloudy. And 0.2, maybe still bacterial growing, almost similar with the control. The next one still growing, but a little bit less. And then this one a little bit less, a little bit less. This is a little bit less. And finally over here, no bacterial growth. It means that bacteria will be killed by this 6.4 dose. And then of course, the 12.8 will be no bacterial growth because this is higher dose. So based on this information, then we know this is actually the minimum concentration that will kill the bacteria. So the MIC from this test will be 0.4 milligram per liter. So 0.4 dose will be the one that will be used, for example, for development of drugs with this dose. Okay, so this is how to test for the MIC. Okay, so remember MIC, this is done outside human body. It's called the in vitro test. Okay, so what happens if it is in vivo? It's maybe a little bit different. Therefore, we have to know whether that drugs, if it is given to human body, will be safe or not by calculating the what we call the therapeutics index. So we basically have to also perform the test inside the cell, inside the human body. Let's say, after the test, we perform the MIC test. Let's say we have 3 different drugs, maybe A, B, C. Okay. So this drugs is used for this, the same type of infection. Let's say when we test the MIC, okay, for drug A, let's say we have 1 milligram per liter. Okay, the MIC is 1 milligram per liter. And when we test B, it's actually also 1 milligram per liter. And C also 1 milligram per liter. Okay? So this is the MIC. And then we also perform toxicity tests to human body. So toxicity tests. Okay. Let's say drugs A actually causing toxicity when it is reaching 10 milligram per liter. Okay. Drug B, it causing toxicity when it is reaching 1000 milligram per liter. Okay, it means that it is safer because it's given toxicity to human body at higher concentration. Let's say C is actually giving the toxicity to human cell at 100 milligram per liter. Okay, so based on this test, the MIC test and toxicity test, we actually able to calculate the TI, okay, the therapeutic index. Okay, so the TI for A will be, so we actually divide the toxicity against the MIC. So A will be 10 divided by 1. It's going to be 10. So the index will be 10. B will be 1000 divided by 1, by the MIC. And then the result will be 1000. And C It's going to be 100 divided by the MIC, which is 1. Then the result will be 100 Ti. Now, from this information, remember the high index is desirable because high index means it's actually safer. So based on this information, then the best drug to be used will be the B antibiotics, okay, the B drugs, because what? The TI is 1000. So the higher the index, the better the drug's gonna be, more efficient and also safer to human body. I think that's all for chapter 12. We finished chapter 12. Uh, we will see you again on the next chapter, which is chapter 13. Goodbye now.
Hi everyone, welcome to Medical Microbiology. Today we will discuss chapter 13, Microbes and Human Interaction. This chapter will be divided into 3 lectures. In the first lecture, we will introduce about the microbes and human interaction. Okay, just everybody know that we are actually not alone. This means that in our body, we actually have a lot of interaction with microbes, microorganisms, including bacteria, fungi, or maybe some viruses. Some of them we call as the normal flora or indigenous flora, which is the flora or the microorganism that live on us, and many of them is actually beneficial for us. They provide protections, okay, against some pathogenic flora or microorganism. Also, some microbes is actually involved in our maturation of our immune system. So the T cell, B cells, they need to be mature, and in order to be mature, sometimes they get the challenge from these microorganisms. The problem is some of the microbes actually can invade the sterile tissue, and this is the one that will cause the disease to us. Okay, again, in our body, we have interaction with some microorganism, and this microorganism usually called as a normal resident microbiota, indigenous flora, microflora, normal flora, or commensalism flora. So these are different names for microorganisms that live on us, and most of them are beneficial for our immune system, beneficial for us. And then there are some of them that causing infection. Okay, it is called a pathogen. So pathogen is the microorganism that cause infection to us. So the infection is actually the condition in which pathogenic microbes penetrate the host defense, enter the sterile tissue, and then multiply in that tissue or in the organ. And then infectious disease is the infection that is caused by this pathogen. And they can cause disruption to the tissue and the organ. So again, infectious disease is the disease that caused by pathogens. So again, we actually have contact with microorganisms, Okay, so initial contact over here. Some of them colonize, okay, in our system, and many of them is actually beneficial. This is what we call the normal flora or indigenous flora. But some of them can invade our sterile tissue, and this will cause the infection. So again, infection is happening in the sterile tissue that is invaded by microorganisms. And okay, the one that causing infections, it will challenge our immune system. If our immune system able to take care of this pathogenic flora, then we will develop what we call the immunity. Okay, so they actually also can make our immune system become stronger and better. However, if this pathogenic flora infects our system and our immune system unable to protect us against this pathogenic flora, then it might cause what we call the morbidity or disease, or even mortality or death. Okay, so the disease or death occur when our immune system unable to fight against the infection. Okay, so again, this development of immune system can be beneficial for us, but on the other side, the pathogen can have the adverse effects into our body, which is causing disease or morbidity, or even causing death or mortality. This diagram just showing you how this organism can enter into our body. So usually they use one of what we call the portal of entry. In this case, using the respiratory system, which is our nasal cavity. So enter into our body in this nasal cavity, then it can go to the sterile tissue. Okay, it's causing infection. For example, here what we call the otitis media, okay, and then maybe enter into the layers of protective layer of brain and spinal cord is called the meninges, causing the meningitis, for example. Or if enter into the lungs, can cause pneumonia. So there are several diseases that may be caused by this entrance or invasion of pathogens into our sterile tissues. Again, the resident microbiota is mean they use our body as their habitat. So they live on us. Okay? The area that usually become their habitat is the area of our body that have contact with the outside environment. Like for example, the skin, okay, the upper respiratory tract, okay, the nose, for example, throat, or upper GI tract, like the mouth, okay. And even for the GI tract, even the intestine is considered as the part that also contact with the environment. Therefore, there are also some resident microbiota that live in our intestine, especially the large intestine or colon. There are several types of microbiota. If it is staying only for a short period of time, it is called a transient microbiota. But if they establish and become, the microbes that live for a long time, then they become what we call the resident or resident flora. So table 13.2 is showing us the sterile tissue. It means that the sterile tissue has to be sterile from all type of microorganisms, including the normal flora. Okay, so if the sterile tissue invaded by flora, whether pathogenic flora or the normal flora, then there will be infection. So infection is happening actually in the sterile tissue. Like for example, our heart and circulatory systems are sterile. Okay, no pathogen over there, no microorganism live over there. There is no normal flora live over there. Okay, liver, kidney, and bladder, lungs, brain, and spinal cord, bones, ovary or testis, gland, okay, pancreas, salivary gland, sinuses, middle and inner ear, internal eyes. And these are considered as the sterile site of our body. The fluid, for example, okay, blood, it has to be sterile. The urine, when it is still in the kidney, it will be sterile. Okay, in the ureter and also in the urinary bladder. They are actually sterile fluid. Okay, cerebrospinal fluid and CSF, they are also sterile from microorganisms. Saliva, when it is still in the salivary gland, they are sterile. And semen, also sterile before it is injected from the the male organs. Now, when is actually the first colonizations of normal flora happen? This is actually started when the the baby, during the birth of the baby. Okay, so this is the first time when human actually have the colonizations of normal flora. Okay, so after that there will be additional contribution, which is maybe from the mother, from the father, or from the caregiver, or from the hospital, and also maybe from the food or the drinks that enter into the babies. Okay, so just for information, uterus, it is actually sterile. So whenever the baby born, then he or she will have the first colonization of microorganism or normal flora. Okay. So the resident microbiota or the normal flora, generally they are stable, but sometimes increase or decrease, okay, based on condition of that person. The diet, hygiene, hormones can also affecting the fluctuation of this normal flora. And again, in many cases, this microbiota is actually benefit for the host because they actually protect us against the pathogenic flora. So this is what we call the microbial antagonism or microbial competition. So they compete with other flora, and many of these other flora are actually pathogenic flora. But they can also cause infection if they enter the sterile tissue. And the infection that caused by this normal flora is called the endogenous infection. because the normal flora is actually present on us, and then maybe sometime because of that condition, something happened, that normal flora enter into our sterile tissue, then it will cause infection also. Okay, now the common site of habitat for the indigenous flora and also the types of the normal flora that live on that site. Okay, for example, the first one, of course, the skin. Okay, so skin, this one directly interact with environment, and then there are many microorganisms in the environment. Then some of them will stay and live in our skin. So some of the normal flora of the skin are Pseudomonas, Micrococcus, Corynebacterium, Propriobacterium, and this one is actually can cause acne, okay, Propriobacterium acne. So one time this normal flora, can cause the acne to the host. Staphylococcus and Streptococcus. So these are the types of normal flora that live in our skin. And then for the fungi, okay, so like Candida, Malassezia, and Rhodotorula, these are a normal flora or fungi that usually also live in our skin. For the arthropods, this is actually animal, Demodex mite is an example of some insect. Okay, this is actually lice that live on human skin. And then for GI tract, okay, for GI tract, there are several locations. If it is the mouth or the oral cavity, so the bacteria that live in this cavity mostly Streptococcus, Neisseria, Philonella, Fusobacterium, Lactobacillus, Bacteroides, Actinomyces, Echinella, Treponema, and Haemophilus. So this is a bacteria that usually live in oral cavity or in the mouth. Fungi, mostly Candida species, and for the protozoa, there is also an Amoeba gingivars. So this is in the mouth. Okay, GI tract. Another location is in the intestine, especially large intestine. So another name of large intestine is colon. Okay, examples of bacteria that live in this colon are Bacteroides, Fusobacterium, okay, Clostridium, Lactobacillus, E. coli. Okay, the name is telling them. So this bacteria live in colon. Fungi like Candida, protozoa maybe in Amoeba coli. Okay, again, name is telling you the question. Trichomonas hominis. For the respiratory tract, it will be the upper respiratory tract, like the nose and the throat. Okay, because lower respiratory tract, like lungs, it has to be sterile. Okay, so upper respiratory tract, and there are some bacteria that also live in this area. And then for the genital tract, okay, bacteria that live in this area mostly Lactobacillus, Streptococcus, and then for the fungi mostly Candida. For the urinary tract, bacteria that live normal bacteria that live here, mostly Staphylococcus, Streptococcus, Lactobacillus, eyes. It's the eyes, also face direct to the environment. So therefore, there are some bacteria that also live on our eyes, like Staphylococcus, Streptococcus, Neisseria. And the ear, of course, this should be the outer ear, because the inner ear and the middle ear should be sterile. Okay, so the one that is live in outer ear, mostly Staphylococcus bacteria. For fungi, sometimes Aspergillus, Penicillium, Candida, and yeast. So this is the site that usually normal flora live. Again, for the first site, which is the skin, okay, mostly this normal flora will live on the top layer of the skin, which is the epidermis. Some of them maybe penetrate a little bit to the dermis area. Okay, so the one that live stable in this area is called the resident flora. The one that just live for short time, okay, sometimes they increase the number, sometimes they actually out from the area, is called the transient normal flora. Okay, GI tract. And the whole GI tract is actually face to the environment directly. Yeah, just remember when we eat, when we drink, the food and the drink that we take is actually carry a lot of bacteria. So therefore, the whole GI tract organ over here, starting from the mouth, the throat, the esophagus, stomach, intestine, rectum, and anus, they're all actually facing to the environment. Then they usually also have normal flora that live on this side. So they become habitat for some normal flora. Therefore, again, mostly the oral cavity will have a lot of normal flora that stay in this area, and also the large intestine, and this is the second organs of GI tract that also have many normal flora that live on it. Okay, for the mouth, there are many of them. Okay, especially if we don't really clean our mouth, our teeth, then there will be a lot of normal flora or microorganisms that live in this area. So many of them in the genus of Streptococcus, like Streptococcus sanguis, Streptococcus salivarius, Streptococcus mitis. So this is a type of bacteria that usually live in our mouth. In the second part of GI tract that usually have a lot of normal flora or microorganism is the large intestine. And examples of bacteria that live in this place is Bacteroides, Bifidobacterium, Fusobacterium, Clostridium. Okay? So these are examples of bacteria that usually live in this place. Respiratory tract, okay, and again, mostly on the upper, okay, upper respiratory tract. Okay, that's the area that usually have a lot of normal flora. The lower respiratory tract over here, they are sterile. Okay? So if there are flora that live in the sterile tissue, then we have infection. Okay. So again, most of the normal flora will stay in the upper respiratory tract, like for example nasal cavity, mostly Staphylococcus, Streptococcus bacteria, Neisseria bacteria. For the genitourinary tract, okay, so mostly against in the lower part. Okay, like for example, for the female side will be around this. Okay, like vagina, urethra. Okay, that's the place where some normal flora maybe live. For the male, it's the same. Maybe the lower part over here, which is mostly in the urethra. Okay? And the upper part, like for example, bladder, kidney, testicle, there will be they are sterile. Okay, so the same thing with this uterus, for example. Okay, kidney, bladder, they are also sterile tissue. So no microorganism live in the sterile tissue. If they infect the sterile tissue, then we have the infection. And again, normal flora is many of them is actually essential to us. They protect us against some pathogenic flora. So this is the reason why there is an idea to put normal flora in the diet. It's called the probiotics. It is to increase or to balance normal flora, especially in the large intestine. Okay, so this is the introduction of normal flora back into the body. Okay, this is the development of infection. Again, the infection is happening when the flora, whether normal flora or pathogenic flora, enter into the sterile tissue. The first thing, okay, they have to find the portal of entry. And again, portal of entry will be organs that contact with the environment directly, like skin, okay, GI tract, upper respiratory tract, urogenital tract, okay, and some indigenous biota actually also able to enter to the sterile tissue and cause infection. And then after finding the entry, okay, the portal of entry, then they have to attach to this site. Okay, so remember, virus, they use spike to attach to the host cell. Bacteria, they use fimbriae, sometimes capsule, glycocalyx. Okay, yeah, this is viral spike. Worm, for example, they have hook on their head, yeah, to attach to the tissue or to the cell. And then after they attach to that site or the cell, then they have to be able to survive against the host defense mechanism. Okay, so some bacteria, for example, have capsule that make them able to avoid the phagocytosis, killing by the white blood cell. Okay, and also, yeah, some of them able to have some action that make them, live or live fighting against the host immune system. After surviving the host defense, then they have the ability to cause damage to the host tissue and then causing the disease. Okay, they actually produce what we call the virulent factors. Okay, so virulent factors are factors, mostly chemicals, that produced by the microorganisms to damage the host tissue. So some virulent factors like toxin, so some bacteria or microorganism actually produce toxin to damage the host tissue. Some bacteria or microorganism also produce enzymes that can damage the host tissue. And some of them also produce chemical or use chemical to causing lysis, uh, like for example lysis on the host red blood cell. So it's actually break the red blood cell. And then they also can cause the indirect damage to the host tissue. And then some of them, yeah, so they stay inside the cell. They also can exit from the host tissue, okay? And then when they exit, they actually will be able to transfer to the next uh, host, okay? So they actually, when they exit the host tissue, they actually able to transfer to other hosts, maybe either human or maybe animals. Okay, so the portal of exit will be very similar with the portal of entry, like for example, respiratory tract, skin scale, okay, so uh, feces, okay, urogenital tract, like urine. So some urine maybe carry some microorganism, and then if we enter into the system, onto the environment, it might be able to transfer to other, or maybe through blood. Okay, so blood that has microorganism or disease on it can also be transferred to other hosts. This is some major factors in development of infection. Okay, first the age. So all age or extremely youth, like a baby, for example, or premature, most of them are having less immune system. I mean, their immune system for the baby usually is not developed yet. For the old people, their immune system is become weaker. So this is weaker immune system. Therefore, they usually have the higher risk to get infection. Genetic defects in immunity, okay, like people with AIDS, for example, then AIDS people usually will have higher risk to get the infection. Surgery and organ transplant, this one also can increase the risk of getting the infection. Organ disease like cancer, liver malfunction, and diabetes will also increase the risk of having infection. Chemotherapy, people with cancer sometimes they have chemotherapy, and these chemotherapies most of the time will decrease the immune system. So people with the chemotherapy usually also have the higher risk to get the infection. Physical and mental stress. This is also something that can increase the risk of getting infection. And other infection can also causing the in other infection will come and causing the higher risk of getting a new infection. Okay, for the pathogens, okay, remember we have the normal flora, which is mostly benefit us, and the pathogenic flora, which is the microorganism that can cause disease to human. Okay, so the pathogens can be divided into two types. The first one is called the true pathogen. So true pathogen is microorganism then that capable of causing disease in healthy persons. Like for example, flu. So when the flu virus enter into human body, and it doesn't matter whether this human body is healthy or unhealthy, this flu virus can cause infection to him. Okay, so it able to cause disease in healthy person. This is what we call the true pathogen. The second type of pathogen is called the opportunistic pathogen. Mostly they are normal flora. Okay? So this normal flora usually will not cause disease to healthy person, but it might cause disease to people with lower or compromised immune system. Like for example, people with AIDS, they usually can get the infection easier compared to the healthy person. So some organism like maybe Streptococcus, okay, or E. coli maybe will not cause disease to a healthy human, but it might cause disease to people with AIDS because people with AIDS they actually have lower immune system. So the organism that can cause disease, okay, to people with weaker immune system but will not cause disease to normal healthy human, it is called a opportunistic pathogen. And then virulent factors, again, this is the factor that produced by the microorganism that damage the host cell and causing the disease. Mostly these virulent factors are enzymes and toxin. Okay, there are some other type maybe, but mostly enzyme and toxins. These are the virulent factors that produced by the organism, microorganism. I think that's all for lecture number 1. So we will meet again on lecture number 2, which is stages of uh, infection. Goodbye now.
Hi everyone, welcome to Medical Microbiology. Today we will continue chapter 13. This is lecture 2. We will discuss about the clinical stages of infection. First of all, we have to understand that there are three main sequences of infection. The first is finding the local or the portal of entry. So finding the portal of entry. So in this stage, the microbes, especially the pathogenic uh, microorganisms, will try to find the organs or maybe the tissue where they will be able to enter into the sterile tissue of the host. Usually the portal of entry are the skin, respiratory tract organs, GI tract organs, urogenital tract organs, and sometimes there are also placental entry. After finding the portal of entry, then the microbes will start to attach. Okay, so the second sequence will be attachment. Just remember, most viruses, usually the one that have envelope, they will use spike for attachment. Uh, Bacteria, they usually use fimbriae for attachment. Some of them maybe use the slime layer or glycocalyx, okay, for attachment. Some protozoa, they usually use flagella and cilia for attachment. For worms, they have special structure to attach, which is hooks, or maybe suckers, or maybe barbs. So these are the attachment structure of microbes. The third, after they attach to the organs, to the host tissue, then they start the infection. Okay, so the infection, they usually produce what we call the virulent factors. These are usually enzyme and toxin that will break the host tissue, and also they will destroy the host cell, and then the bacteria or viruses will be entering into the sterile tissue and then reproduce in this host tissue. So these are the common sequence, okay, which is the three main sequence of infection. We start with the stage number one, which is finding the portal of entry. Now, there are two main type of microorganisms that can cause disease. Okay? The first one is called the exogenous agents, which is this is the microbes that originated from outside the body. So this is not part of human normal flora. It is not part of the human indigenous flora. So it is mostly coming from outside, like for example, flu. Okay, flu viruses is not part of our indigenous flora. So it is actually coming from outside and then enter into the cell tissue and then cause disease. The second one is called the endogenous agent. This is actually the normal flora that already exists on our body, maybe on the skin. Okay, so whenever they enter into the sterile tissue, then they can cause disease. So this endogenous agent is actually part of our normal flora that enter into sterile tissue and cause disease. Now, the main portal of entry are, of course, the skin. So these actually organs that face directly to the environment, like the skin. So the skin is actually the biggest organs of human body, and this is the place where many microbes will be able to attach and also enter into the host cell, the sterile tissue. The second portal of entry will be gastrointestinal tracts, like the mouth, okay, esophagus, stomach, okay, intestine, rectum, anus. These are organs that face directly into the environment. Maybe the intestine inside our body, but it is actually also facing the environment, which is the food and the drink that enter into the GI tract. So this food and drink actually coming from outside, and they also carry some microorganism and then enter into our GI tract. The next ones, okay, will be the respiratory tract. This is especially the okay, upper respiratory tract, like the nose and uh, pharynx. Okay, this is the most part of the respiratory tract that become the portal of entry of some microorganism. The lower respiratory tract, they are usually sterile organs, like for example, lungs, the alveoli, they all have to be sterile. Okay? So only the upper respiratory tract is considered as the portal of entry for microorganisms. The next one will be the urogenital tract. Okay. This is mostly the lower part of the urogenital tract, like for example the urethra, the vagina. This is part of the entry for microorganism. The upper urogenital tract, which is inside our body, like for example the uterus, cervix, ovary, testicles, bladder, kidney, they are all sterile tissue. Okay, there should be no microorganism in this sterile tissue. So only lower urogenital tract, like the urethra, the vagina, will be the portal of entry for microorganisms. Some other organisms, like for example toxoplasma, they able to use the placenta as the portal of entry from mother to the embryo inside the mother womb. So these are the portal of entry for microorganism. This is just some information about the most common type of microbes that use the portal of entry, especially the urogenital tract. Okay? And these are considered as the STI or STD, sexual transmitted infection or sexual transmitted disease. The most common one is actually human papilloma viruses, HPV. Yeah? So about 14 million cases per year, just in the United States. So 14 million cases of HPV infection through the urogenital tract. And just for your information, the HPV can cause or can initiate the cervical cancer, especially for women. That's the reason why there is a vaccine. If you are older, like maybe 18, 19, you actually have to take the HPV vaccine, okay, to uh, try to prevent against the disease and also prevent against the cancer. The second one is trichomoniasis. It's about 2,230,000 cases per year. Chlamydia, about 1,360,000. HIV, for example, is about 35,000 new infection per year just in the United States. So you can check this list, you can read this list for your information. Okay, the one that use placenta, they usually have abbreviation of STORCH, STORCH. S is for syphilis. Okay, so this is one of the disease. This is actually caused by bacteria that can be transferred to the embryo from the mother. Toxoplasmosis. This is also a disease that can be transferred through the placenta. O is for other disease like hepatitis B, AIDS, and chlamydia. So these are the diseases that can be transferred through the placenta. And then R is for rubella. C is for the cytomegalovirus. And H is for the herpes simplex viruses, especially the
SHV number 2, the genital herpes. So these are the diseases that usually transfer through the placenta. Now, when we talk about the infection, then we have to know the ID, which is the infectious dose of the pathogenic microorganism. So the ID is actually the minimum number. So this is the lowest number of virus or maybe bacterial cell that can start the infection. So therefore, the lower the ID, then the greater the virulent factors, and also the greater is the it's called a contagious disease. Okay, so the smaller the ID, the more contagious the disease is gonna be, or the pathogen is gonna be. So look at this table. Okay, so measles, this is a virus. Only one virus actually can start the infection, and it is use the respiratory tract as the portal of entry. So this measles is considered as the most contagious disease, because one virus able to start the infection. Another one that is also very contagious is shigellosis. 1 to 10 cell through the ingestion can start the infection. Compare, for example, with the cholera over here. So cholera, it requires about 100 million cells to start the infection. So if we compare with the measles and cholera, then measles is very, very, very contagious, easy to be transferred, and easy to start the infection. Compared to the cholera, it's require at least 100 million cells to start the infection. So the lower the ID, the easier of these pathogenic microbes to start the infection, and usually they are more contagious. Okay, now the second stage, which is the attachment or the adhesion. Again, this is where when the microbes start to attach, find the right cell to attach, and then after attachment they will start to do the infection. So just remember again, virus, they usually use spike for attachment. Bacteria, they use fimbriae, and also glycocalyx, for attachment. Protozoa, they use flagella, cilia for attachment. Worms, they have special structure like suckers, hooks, and barbs to attach to the host tissue. Okay, so these are examples of microbes and their attachment structure and where that structure attached. So the target cell for that attachment. For example, the Neisseria gonorrhoeae, this is a bacteria that can cause disease called the gonorrhoeae. They have pili. Okay, so pili for attachment, usually on the genital epithelial cell, so in the genital area, urogenital tract. E. coli, and the name should tell you that this bacteria will attach to the colon or large intestine. It can cause diarrhea,
And then what they use? They use fimbriae to attach to the intestinal cell, especially to the large intestinal cells. Shigella and Salmonella, they can cause disease called the gastroenteritis. They use fimbriae for attachment into the intestinal epithelium, especially the small intestine. Treponema, this is another bacteria that can cause disease called the syphilis. They have hook. Okay, so these treponema have hook that attach to the host cell. Usually they use the urogenital tract organ as the portal of entry. Trypanosoma, this is protozoa. Okay, so this is a type of protozoa. They can cause disease called the trypanosomiasis. And what they use flagella or flagellum to enter into the host cell. Many trypanosoma actually red enter into red blood cell and destroy the red blood cell. So mostly they can cause anemia because they perform the distractions of red blood cell. So they use flagella to enter into this red blood cell. Okay, the next stage, which is infection. Again, they use what we call the virulent factor. Okay, so the virulent factors, usually enzymes and toxin. Now, what's the difference between enzymes and toxin? So this enzyme, they usually destroy the area between the cell. Right? The area is called the interstitial cell. They break the junction between the cell, and then when this junction between the cells is broken, then this bacteria will be able to enter into the sterile tissue. So the way the enzymes produce usually is produced in the interstitial fluid or area, and then they break the junction between the cell. Like for example, if this one has skin cell, they break that junction between the skin cell, and then when it is broken, bacteria will enter into the sterile tissue. Okay, that's the enzyme. The toxin, okay, they're also produced by bacteria, but they actually enter into the cell. So the cell is become infected, and then usually it will be destroyed. So the toxin mostly will destroy the infected cell, while the enzyme usually break the joint or the junction between cells. There is another virulent factors that some bacteria have, not all bacteria. Okay? It is called the capsule. So bacteria that have capsule, like this bacteria, for example, they're able to protect themselves against phagocyte. So phagocyte is a type of white blood cell that fight infection. So you can imagine if this white blood cell unable to kill the bacteria that have capsule, then this bacteria will be able to stay in the tissue, will be enter into the sterile tissue, and will be able to reproduce in the sterile tissue. So these are some example of virulent factors of microorganisms that they use for infection. The next one, okay, some bacteria, for example, Salmonella, they produce special proteins that actually break the intestinal cell. So whenever this intestinal cell is broken, then this salmonella will be able to enter into the cell and stay inside the intestinal cell. And then inside the intestinal cell, they start to reproduce. They will also destroy this intestinal cell, then usually causing bleeding. So salmonella usually causing diarrhea that accompany with bleeding. Okay, so this is example of uh, infection factors or filtration factors of some bacteria, microorganisms. Okay, now we're gonna talk a little bit about toxin. So this toxin again is produced by some microorganism, and even it's also some of them is produced by uh, microorganism. Yeah, like for example, snake they produce the toxin like poison. Okay, even plant they also produce toxin. So the one that we focus is the toxin that produced by microorganism like bacteria. Yeah? They produce also toxin that able to destroy the host cell. The adverse effect of the toxin is called toxinosis. So this is the adverse effect of toxin. Toxemias, when the toxin actually spread from one organ to another organ through the blood circulation. So this is what we call the toxemia. And then intoxication, this is especially when it is entered through the ingestion. Like for example, people eat uh, food that is actually contaminated with poison or toxin, then it is we call as the intoxication. So just remember intoxication related to food or uh, beverages or drink consumption. Okay, there are two types of bacterial toxin. The first one is called the endotoxin. So endotoxin is actually produced by bacteria that are destroyed. So like for example, white blood cell destroy this bacteria. Boom. When this bacteria is destroyed, they will release the endotoxin. Okay, so basically endotoxin is produced by dead bacteria. Okay, and the exotoxin, it is produced by living bacteria. So these living bacteria able to produce the endotoxin, exotoxin. So this is living bacteria. Okay, so the endotoxin eh, is produced only by gram negative bacteria. So only gram negative bacteria have the ability to produce the endotoxin when they are destroyed. Right? The exotoxin is produced by both gram negative and gram positive bacteria. Okay, so exotoxin produced by both gram positive and negative bacteria, and they are produced by live cell. Okay, so we compare between exotoxin and endotoxin. So this one actually exotoxin over here. Okay, so again, it's produced by living bacteria. Okay, so it's produced this exotoxin, and usually they will destroy the cells, the host cell, especially the heart, muscle cell, blood cell, intestinal tract cells. So these are the exotoxin produced by living bacteria and destroy some of the host cell. Now the endotoxin, this is the endotoxin. Just remember again, it is produced by dead. This is dead bacteria. So when the bacteria are destroyed, then they can release the endotoxin. Usually the effects are causing the what we call the physiological effects like fever, malaise, itches, and shock. So compare with the exotoxin, they usually damage the organ directly. And again, the exotoxin, they are produced by living cell, okay, of gram positive and gram negative bacteria. Okay, so this is exotoxin. Now the endotoxin is produced by dead cell of gram negative bacteria. Okay, so these are the difference between an exotoxin and endotoxin. Again, you can read the list of these, the table of the different differences between exotoxin and endotoxin. So toxicity, so the exotoxin actually very toxic in small amount, okay, while the endotoxin it require higher dose or higher concentration. The effects on the body, just remember they usually destroy the cell for the exotoxin, blood, liver, nerve, skin cell. The endotoxin usually causing the systemic and less specific symptom, like for example, fever, inflammation, weakness, shock is a very general symptom. And then the chemical composition for the exotoxin is small proteins, while this one is actually lipopolysaccharide of the cell wall. And just remember, this lipopolysaccharide is only present uh, on the cell wall of gram negative bacteria. So therefore, it's only produced by the gram negative bacteria. The exotoxin usually unstable, endotoxin is very stable against heat. Toxoid formation, yes. So exotoxin can be converted into toxoid. So toxoid is actually a weak type of toxin that can be used as vaccine. Okay, so vaccine can be also made of toxin. It's called the toxoid. The endotoxin, it cannot be converted into toxoid. Fever stimulation usually not for the exotoxin, yes for the endotoxin. Manner of release, again, this exotoxin produced by the live cell, while the endotoxin is produced by cell that is destroyed. lice mean destroy or cut. So endotoxin is only produced by that bacteria, gram-negative bacteria. And the typical source, again, for the exotoxin produced by the gram-positive and also gram-negative bacteria. Endotoxin is only produced by the gram-negative bacteria. Example of disease that caused by exotoxin are tetanus, diphtheria, cholera, and anthrax. These are some examples of disease caused by the exotoxin. For the endotoxin, examples are meningitis, endotoxic shock, and salmonellosis. This is the one that caused by the endotoxin. Now we're gonna go to the process of infection, or it is called the clinical stage. Okay, so clinical stage of infection. There are four of them. The first one is called the incubation period. This is the time, okay, from initial contact, okay, to the appearance of the first symptom. So this is what we call the incubation period. Different organism will have different incubation period. Like for example, COVID-19, maybe the incubation period is between 2 to 5 days. Okay, but for the HIV or AIDS, the incubation period can be 2 until 15 years. So you can imagine that. So much different. Eh? So usually during the incubation period, eh, there will be no sign, no symptom. Okay, it means that the patients actually carry or have the disease, but he does not realize it because there is no sign and there is no symptom of the disease. That's during the incubation period. And they still be able to transfer to other people. Okay? So sometimes it is very dangerous because they don't feel the sign and symptom, but they're able to transfer this pathogenic organism to others. Okay? The second stage will be the prodromal stage. Usually, okay, this is the start of symptom and sign, but the symptom is very non-specific. Okay, it is what we call the general symptom. Okay, such as fever, weakness. Okay, so feeling discomfort, for example, this is very general symptom. We don't really know what caused this general symptom, general discomfort. Okay, so therefore, this is the stage is called the prodromal stage. After the prodromal stage, then there will be period of infection. This is the time when the organism is actually multiplies in the high level. So they multiply so much, and then it will give the specific sign and symptom for that specific disease. So we will know what type of disease is this because it has very specific sign and symptom. Okay, it's not only headache or maybe fever, but there will be more specific symptom of this disease. This is what we call the period of infection. And then after that, if our immune system, the white blood cell, the antibodies able to fight against this disease, Then we will have what we call the convalescent period. This is actually the period of recovery. So we feel better. Eh, But we actually still have microbes in our body, and we still be able to transfer to others. So this is another uh, stages that sometimes is noticeable, but it can transfer the disease from uh, one person to another person, just like the incubation over here. So the incubation, there is no sign or symptom, but the patient still be able to transfer to others. And the recovery period, maybe the patient feel better, and the things is okay, but he actually still be able to transfer to others. Okay, so it is in the uh, picture. So this is the picture of stages of clinical infection. Again, start with the incubation period. And again, this is depend on the pathogenic organism. For flu, COVID-19, for example, usually the incubation period is between 2 to 5 days. Okay, but for HIV, okay, maybe 2 to 15 years. So the incubation periods are longer for HIV. Okay, let's say we're gonna put this COVID-19 over here, and this is for HIV. Okay, now the again during the incubation period there is no sign, no symptom. So usually no sign or symptom. Okay, the next one is prodromal stage. This will be general symptom. Okay, so over here, general symptom. Okay. Maybe for COVID-19 it's around 2 to 5 days. Okay. For the HIV maybe several years. I mean several maybe days or months. Okay. And then the next stage will be the period of infection. And this is when the bacteria or viruses reproduce in high number. So there will be specific sign and symptom. Yeah. So for the HIV, usually take about 7 to 10 days. Okay, this is when our immune system, the antibodies, the white blood cell fight the infection, and we feel that, and we have the sign and symptoms for that disease. Okay. For HIV, maybe it takes years. So this during this infection period. And then after that, if our body able to protect us again or fight against this disease, then will be recovery. So the symptom and the signs will be reduced. Okay, so the disease is actually gone. So it's a recovery period for the COVID-19, maybe about 5 to 7 days. Okay, so total the whole things, okay, from the incubation period until the convalescent period, usually for COVID-19 is about 10 until 21 days. So this is the time when, if you have COVID-19, you have to stay away for other support, at least, okay, minimum 10 until 21. days. Okay, so these are the clinical stages of infection. Okay. I think we're done. We will see again on the next lecture. We will talk about pattern of infection and epidemiology. Goodbye. Hi everyone, welcome to medical microbiology. Today we will continue chapter 13. So we will have lecture number 3, which is the last lecture for this chapter. We will discuss about the pattern of infection and epidemiology. Okay, so pattern of infection. So if you see the infections, maybe the infection is local. Yeah, it's called the localized infection, just in specific area of the body, okay, and then causing the infections only on that specific area or organs. The second pattern of infection is called the systemic. This is when the pathogenic organism is actually spread from one area to another area, usually using the cardiovascular system, the blood circulation. So when the pathogenic flora enter into the bloodstream, then they can cause what we call the systemic infection. The next one is called the focal infection. This is the transfer of one infection in one area, in one local infection, okay, and then going to another local infection. Okay, so therefore it's called a focal infection. They might be carried by different way of transfer, not really through the blood circulation. Okay, like for example, when you have a skin infection, then you scratch that skin infection and you put it in the mouth or maybe in the genital area, and that infection transfer to the new local place. So this is what we call the focal infection. If the infection is mix of many disease or microbes, then that place is called has the mixed type of infection or polymicrobial infection. So poly means many, microbia means microbes. So the infection that is caused by multiple bacteria, many bacteria. Now, according to the sequence of occurrence, then there will be primary infection and secondary infection. So primary infections mean the first local area or organ that has infection. The secondary infection is when after the primary infection, that infection go to another place and causing the secondary infection. Based on the intensity, okay, there may be acute infection, which is very rapid and usually short life effects. Like for example, flu, it is a very rapid infection, maybe only 7-10 days, but it's break a lot of tissue and cell. Okay, so it's very rapid production, but it's very short life, like between 7 to 10 days. The chronic infection, okay, this is the infection that progress for years, stay for years in a host cell. Example is TB, for example, tuberculosis. So take that disease stay in the tissue, in this host cell for years. Okay, so again, this is just the figures of pattern of infection. Like for example, this is local infection or localized infection. This is systemic because it is carried by the blood circulation. This is a focal infection from one local to another local infection. This is type of mixed infection because this is caused by many bacteria. Right? This is secondary and primary infection. So primary infection, for example, maybe in the urethra, okay, urinary infection. It can go to another place, which is the vagina, for example, as a secondary infection. So this comes first, and then the second one follow. So this is the primary and secondary infection. Now, this is really important for us, right? About sign and symptom. So you make sure you know that sign is any, okay, evidence of infection or disease that can be observed, right, or measured. It can be observed and measured. Like for example, fever. So you can measure the fever. Like for example, you can use the thermometer if it is 0.104 degree, for example, then it is fever. So you can measure it. So normal body temperature is around 96 to 98, right? So when it is fever, it increase into more than that, maybe 100, 101, 102, and etc. So this is what we call the sign. Right? So the evidence can be observed. Right? The symptom of infection, this is very subjective evidence, which is we cannot measure it. It is only sensed by the patient. Okay? Example is headache or itch or pain, for example. So you cannot measure this. Therefore, this is what we call the symptom. So symptom cannot be measured. Sign, it can be measured. So these are the list of sign and symptom. So make sure you're able to distinguish between these sign and symptoms. So fever, right? Of course, this is the sign because you can measure fever. Right? You can use thermometer, right? Thermometer to measure the fever. Chill, you cannot measure chill. Right? Septicemia is mean that you can take the blood sample and see under the microscope and see whether there is a bacteria or not in the blood. Yeah, septic means bacteria, emia is blood. So the presence of bacteria or the presence of microbes in the blood. So you can measure it, right? You can also measure the microbes in tissue fluid. You can observe the abnormal chest sound, okay, so love depth sound, either faster or slower. So you can measure it. This is sign. Skin eruption, you can see it, okay, so this is a sign. You can measure it, you can observe it. This is sign. Leukocytosis mean, okay, the measurement of white blood cell. So this is white blood cell become reduced. Okay, so reduction of, sorry, this is increase. So cytosis means increase. So the increase of white blood cell. Leukopenia is actually decrease number of white blood cell. And again, you can measure it. You can take a sample, blood sample, and measure it. Swollen lymph nodes, of course, you can see the swelling. Abscesses, you can see the skin abscesses, for example. Tachycardia, this is increased heart rate, you can measure it. Okay? You can measure the antibodies in the serum. Okay? So these are the signs. So anything that can be observed, eh, observed or measured, this is the sign. Symptom, so you have to ask. You have to ask the patient because only patient know if they have this condition or not, like chill, pain, irritation. You cannot really measure them. Nausea, fatigue, malaise, chest tightness, itchy, headache, weakness, abdominal cramp, anorexia, which is lack of appetite, and sore throat. You cannot really measure them. So if something that you cannot measure, then it considered as a symptom. Okay? So physical sore, this is a sore throat is when the sore, the feeling of sore, we cannot really measure it, but you can measure the maybe the redness. Okay? So when we talk about the uh, throat infection, for example, strep throat, uh, then you can measure it by looking to the throat, if it is redness, etc. That's a sign. But the feeling, the sore, that's the symptom. Okay, and then usually in disease, you start with the process what we call the inflammation. So the inflammation, and it can be sign and symptom. So the sign, if it is sign, again, this is the thing that we can measure. The sign of inflammations are edema, so the swelling. So you can see the swelling on the tissue. That's a sign. Granulomas and abscesses, again, you can observe this. Okay, and then lymphadenitis, again, the swollen lymph node. We can also see or observe this sign. So this is considered as sign. Another sign of infection, we can take the blood samples and then calculate or measure the white blood cell. If it is increased, it's called the leukocytosis. eh. If it is decreased, it's called leukopenia. We can also check whether there is microorganisms in the blood or not. And that process is called the septicemia, or that sign is called the septicemia. If it is bacteria, then it is called the bacteremia, bacteria in the blood. If it is virus, then it's called the viremia, which is virus in the blood. Okay, now there are some infection that unnoticed. It's called the asymptomatic infection. Okay, so when it is called in asymptomatic, it is actually include the sign also, no symptom and no sign. Okay, like for example, during the incubation period, mostly the person that have the microorganism will not notice because there is what? There is no sign and symptom. Then it is what we call the asymptomatic infection. Okay. Now the portal of exit is very similar with the portal of entry. Like for example, if they enter to the respiratory tract, they will also exit to the respiratory tract. Like for example, through the respiration, maybe breathing, coughing, sneezing, that's the exit. Okay? Or maybe through the salivary portals, okay, like saliva. Okay? They also carry some microorganism. Okay? If it is enter into the skin, okay, using the skin as a portal of entry, they can also use the skin as a portal of exit. Like for example, during the slough of the skin and the scalp, there are some microorganisms that will be also carried out from the skin. If it is enter into GI tract, okay, mouth, intestine, they usually also exit through the GI tract, like for example using the fecal exit, the feces. Okay? If it is urogenital tract, so they use urogenital tract as a portal of entry, then they also usually use the urogenital tract as the portal of exit, like for example urination. So urine actually they maybe have some microorganisms in, and uh, remover for blood or bleeding. It also can carry microorganism from uh, the host body tissue. Okay, now we're gonna talk about type of persistence. Persistence of infection. Okay. The first one is called the latency. So this type of infection that usually uh, perform what we call dormancy for many months. Okay, okay. So like for example, herpes virus, they do dormancy for many months, and then at a certain time they can cause the disease, and this is when what we call the outbreak time. Okay, usually you will see the sign and symptom during the outbreak time. Then there will be no sign and symptom during dormancy. So this is what we call the latency type of persistent infection. The second type of persistent infection is chronic. Okay, chronic infection. Usually, the person that have that disease will have that disease and then will have the sign and symptom uh, for the whole time. Okay, so this is the chronic. Like for example, TB, okay, tuberculosis is a type of chronic infection. While for the latency, for example, is herpes. So herpes is a type of latency. Sequela, this is a type of long-term and permanent damage on tissue or organs. So this is a continuous infection, and then that infection will cause the damage to the tissue and organ of the patient. And sequela sometimes is also called as, complication here. Okay, so complication or damage in tissue and organ. Now we're gonna talk a little bit about the pattern of transmissions of disease. It's called the epidemiology, okay, the study of frequency and distribution of disease. This is the epidemiology. Okay, this involves many disciplines, maybe microbiology, anatomy, physiology, immunology, medicine, psychology, sociology, ecology, and statistics. Right? So epidemiology is the study of frequency and distribution of disease. So we also have to consider what we call the reservoir. The reservoir is any condition or any place or habitat, maybe living or non-living, okay, natural habitat that is used by bacteria or viruses to stay. So it could be human, animals, soil, water, land, that can be the reservoir. So any habitats that used by the microbes to stay or to reproduce maybe and then transfer to other people. The source is a specific condition or individual or object from which the infection actually acquired. Okay, so maybe the source and the reservoir are the same. Like for example, strep throat, the reservoir is only human, and that this human that have the strep throat, okay, which is Streptococcus pyogen, can transfer to other human. So in this case, this human is become reservoir and source at the same time. Rabies, for example, maybe the reservoir is the wild mammal, uh, and then that from wild mammal can be transferred to other mammal, and then finally transferred to human. Okay, the reservoir will be the wild animal, and the source will be the animals that has the uh, rabies virus in it. And then it's become the source of infection to human. Okay, now carrier. So when we talk about carrier, this is individual. So this is human. Okay, so carrier is human. So humans that carry the disease or shelter the disease and then transfer to other. There are several types of carrier. The first one is called the asymptomatic carrier, which is humans, okay, that has the disease, but he does not have the sign of symptom of the disease. However, he able to transfer to others. Okay, transfer to others human. Okay, passive carrier. Okay, this human, this individual is actually do not have the disease inside his body, but he is contaminated with the disease, maybe on his clothes. Okay, and then he able to transfer to other people. So mostly in the hospital, for example, nurse, they might transfer the disease from one patient to another patient. And the nurse itself actually do not have that disease in him. Okay. Incubation carriers, this is again when that patient, that person has the microorganism during incubation time, and usually he does not have the symptom and the sign, but she or he able to transfer to others. Okay. Convalescent carriers, remember this is when the patient in the recovery time he feel better, eh, he still be able to carry the disease to others. So therefore it's called the convalescent carriers. The last one is the chronic carrier. Of course, we know this person actually had the disease. We see the sign and symptom of the disease, like TB, for example, and he able to transfer to other people. So this is the carrier. Okay, like for example, this is asymptomatic carrier, the one that carry the microorganism, but he doesn't have any sign and symptom. So she still be able to transfer to others. During the incubation time, so this is incubation carrier, eh, mostly he doesn't have the sign and symptom, but still be able to transfer to others. This is the convalescent carrier, which is she actually recover from the disease, but she still have some microbes in her, so she be able to transfer to others. This one is chronic, so she has the disease with her. We know that she has the disease because there is sign and symptom on her, and she able to transfer to others. This is passive carrier over here, okay, which is the patient, the nurse, okay. So she come from one patient, okay, to another patient over here. The nurse itself do not have the disease, but he able to carry the disease from one person to another person. So this is what we call the passive carrier. Okay, if it is animals, then the carrier is called as a factor. So factor is used, the terminology used for animal, which is animal that carry the disease to human. There are two types. The first one is biological factors, like mosquito, for example. So most of the mosquitoes are biological factor. It means that the disease actually inside, okay, inside the mosquito is part of the mosquito life cycle. So the disease goes to the inside body or of the mosquito. And then, for example, mosquito bite to patient, he will take that pathogenic viruses or maybe bacteria into their body, and then when these mosquitoes bite other people, then it can transfer the disease to others. So this is what we call the biological factor. Mechanical factor, okay? So this is animal that carry the disease, but the pathogens, the microbes actually do not enter into their body. For example, is flies. Okay, so flies usually carry the disease outside their body, like for example, maybe on their wings or maybe on their legs, and then it can transfer the disease from one place to another place. Zoonosis, this is a special term for infection that only transfer from animal to human, like for example, bird flu. This is example of zoonosis. Transfer only from animal to human. It is not from human to human. Another example is rabies. Okay, so rabies is only transferred from animal to human, not human to human. And these are the example of zoonosis. Zoonosis, that is disease that transfer from animal to human. Like for example, rabies usually transfer from wild animals. Okay, all mammals with bite. Yellow fever. This is with mosquito, for example, mosquito bite. Viral fever, wild animal, that is mosquito. Hantavirus, using rodents as the animals that transfer the disease. Influenza, maybe chickens, swine, okay, some of animals. West Nile virus, usually using mosquito. For bacteria, like Rocky Mountain spotted fever, usually carried by ticks, using tick bite. Psittacosis, birds, and leptospirosis, usually using the domestic animals as the source for transfer or infection. Anthrax, sometimes using domestic animal, brucellosis, cattle, sheep, pigs, plague, using rodents, fleas, salmonella, variety of mammal, bird, and rodent, tularemia, rodents, birds, arthropods. Some miscellaneous disease like ringworm, domestic animal, toxoplasmosis, mostly cats, trypanosomiasis, domestic and wild mammals, trichinosis, maybe swine, bells, tapeworm, cattle, swine, fish, scabies, domestic animal. So this is some disease that transfer from animal to human. Now, there is also some terminology that you have to understand about the infectious disease. Eh? Communicable disease is mean contagious. It can be transferred from one person to another person. Okay, if the ID is very small, then it's become more contagious, like measles, for example. Non-communicable disease or infection. This is the disease that is not contagious, cannot be transferred from person to another person. Okay, pattern of transmissions. It may be using direct contacts. Okay, if we have a direct contact with someone that has the disease, we might be able to get the disease. Indirect contact, usually using some intermediate things to transfer to disease, like for example, using what we call the vehicles, like non-living materials, like the door, the cell phone, for example, food, water. Format mean any object, okay, that have been taught by uh, patients, people with the disease. Okay, so this is what we call the format. Okay, like for example, doorknobs, cell phone, okay, or maybe keyboard. If it has been taught by someone that carry the disease, that object is called as formites. Okay, another uh, intermediate may be through the airborne. Right, this is just examples of transfers of disease. Okay, maybe through the direct contacts over here, or maybe indirect. Okay, so direct contact means the contacts with the patient that have the disease. The indirect contact may be through the intermediate object like doorknob, for example, food, okay, air, or maybe some things that is produced by the animals that carry the disease. Okay, now we're gonna discuss a little bit about the epidemiology. Okay, so they usually perform surveillance, they report the incidents, the frequency, and some of patterns of the disease in some area. Okay? And in United States, uh, it has what we call the CDC, which is the Center of Disease Control and Prevention. And it is the headquarters located in Atlanta, Georgia. So this is the place where they report. They take a sample, they do surveillance, and then they give the information about the disease, especially the infectious disease that spread in the United States. Okay? So in order to understand the epidemiology, you have to know some terminology, like for example, prevalence. Prevalence is total number of existing case. The case that already exists, okay, and then spread maybe on other area. So this is what we call the prevalence. If it is a new case, okay, new case like COVID-19, for example, that things or that term is called as an incidence. So incidence only measure the new case in some area. It's not the existing case. That's what we call the incident. If the disease, the infection cause death in the population, that condition is called the mortality rate. How many
death happening from the infection in certain area. That's what we call the mortality. If the infection causing illness, okay, illness or sickness in a population, then it is called as morbidity rate. Okay, so morbidity mean illness or sickness, while mortality mean death in population. And this is just a graph of influenza reported from 1990 to 2014. So this is based on the ages of the patient that contracted the flu. The flu from 1990 to 2014, spread by the whole year. Okay, so this is HIV. Okay, the spread of HIV in the United States by based on the states. Okay, like for example, California, it's about 15.2, Texas 19.8, and this is the rate of HIV infections in the population. 28 in Florida. And DC is very high, about 55.6. Okay, now the patterns. So the terminology for pattern of infection, if it is local, it's called the endemic. So endemic is called as a local spread of disease. Like for example, let's say there is in some area, in some city, they have problem with their drinking water. It is contaminated with some pathogenic organism. So usually that disease is only occur on that city or place. This is what we call the endemics. Okay, endemics is only in local area. If it is spread on many other area with the irregular intervals, then it is called the sporadic. Maybe one in the southern part of United States. There are several incidents on the western part of United States. There is another incident in the eastern part of United States, for example, irregularly. So this is what we call the sporadic. If it is spread on the wider area that usually have a very similar condition, like a weather condition, for example, then it is called the epidemic. Like, for example, maybe disease that only spread in the southern part of the United States. Or maybe like malaria. Malaria is only spread in the whole area, maybe on the African continent, but it is not really spread on other higher area like a northern area. That's what we call the epidemic. Like malaria, usually it is related to the presence of the factors, which is the mosquito on the area. So the area that do not have the specific mosquito that carry the malaria disease, then it will not have the disease. But this disease is really spread in the many countries in the Africa. So therefore, it's become what we call the epidemic. If it is spread to the whole world, it doesn't really matter whether tropical area, subtropical area, or maybe
maybe the area with four seasons like United States, then it is become what we call the pandemic. Example is the one that we have, like COVID-19. It is pandemic. Epidemic may be like malaria. Okay, usually uh, become epidemic. For the endemic, maybe some diarrhea is only spread in local area. So this is what we call the endemic. So endemic mostly local. If it is spread a more wider area, is become epidemic. Okay, so endemic, epidemic. If it is spread the whole world, it's become pandemic. Okay, so sporadic is when the disease spread in one area to another area with the irregular intervals, then it is become what we call the sporadic. Okay, again, if you see this, this is considered as the endemic. It's very local, only several places. When it is spread higher, that is become epidemic. Maybe the whole country of United States have that disease. That's become epidemic in the United States. If it is only present in some area and then another area over here, another area over here, with the irregular interval, this is become sporadic. And when it is spread to the whole world, then it is pandemic. Okay, so the outbreaks, it is considered as a small scale of epidemic in a more limited area. This is based on the point source of epidemic infection, maybe from one single source. The common source of epidemics is actually coming from the same source, and then the epidemic increase over time. It's called the propagated epidemics. Maybe we can see the picture later. Okay. So another term that you have to know is nosocomial infection, which is the infection that is uh, gained when someone stay in the hospital. So the disease that is actually get when someone stay in the hospital is called the nosocomial infection. So this is healthcare associated infection. Good. So this nosocomial disease, they usually can be get from maybe from the surgical procedures, from the equipment that may be contaminated, from the personnel, okay, like nurse and doctors. They also transfer maybe the disease, exposure to drug-resistant microorganism, or maybe the visitors. It's someone that come to the hospital to visit patient. They might also carry the disease to patients in the hospital. Okay, so this is what we call the nosocomial disease. The common nosocomial disease usually come from the surgicals. Okay, this is number one nosocomial disease, mostly coming from this action or condition. Also, Another one is through the respiratory tract, so from the air, from the respiration. GI tracts, maybe also skin, eh, urinary tract, and blood. So this is the source of infection for nosocomial disease. Uh, the bacteria that's usually causing this nosocomial disease, like gram-negative bacteria like E. coli, Klebsiella, Pseudomonas. Gram-positive bacteria like Staphylococcus and Streptococcus bacteria. Maybe fungi like yeast. Uh, through pathogens like TB. So someone that had TB can be transferred to another patient. Salmonella, hepatitis B, influenza virus. It's about 2 to 4 million cases per year in the United States itself, and it actually costs about 90,000 deaths per year. Okay, so these are the nosocomial disease, the disease that is got during hospital stay. Again, if we see this diagram, mostly surgical area and also respiratory tract. This is a kind of number one source of nosocomial disease. The next one will be GI tract, like diarrhea. Next one will be from skin, eh, urinary tract, and blood. Okay, so again, respiratory tract and surgical incisions is considered as number one source for the nosocomial disease. Okay, so remember when the lister, eh, uh, you know, give the idea of doing the preventions, so the sterilizing the hospitals, okay, using the antiseptic technique. Okay, so Joseph Lister is the one that introduced the septic septic technique in hospital setting. So this is some of the precautions that has to be done in the hospital. And this is continue the aseptic technique in the hospital setting in order to prevent against the nosocomial disease. Yeah, so the whole thing is called the universal precautions. So the doctors, the nurse, the administrations, the patients, they have to be following the procedures to protect against this nosocomial disease. And this is what we call the universal precautions. I think that's all for this chapter. So we finished chapter 13. And remember, chapter 11, 12, and 13 will be used for exam number 3. So good luck with your exam number